Department of Biotechnology, since we are supporting many of the activities, those are very much relevant to climate action as well as environment biotechnology. So we have started and incepted to celebrate this day since 2023. And every year we are inviting one of the chief guests from eminent personalities in the nation. And a few of the investigators from department supported projects, those will be showcasing the activities. So I just request to all of you to please switch on the your video so that we can see each other. And as you all are aware, these are our shared responsibilities. I have only a few lines in Hindi, like परिवेश या पर्यावरण जिसे हम बोलते हैं. तो साजी सांस साजी आस साझा साथ साझी बात साझा दिन साझी रात साझी धूप साझी छाव साझे घाव साझे भाव साझा सार साझा संसार साझा स्वभाव साझा प्रभाव. यहाँ हकदारी सबकी है, ये जिम्मेदारी सबकी है, रही परिवेश सुरक्षित अपना ये पहरेदारी सबकी है. इसी के साथ मैं आदरणीय डॉक्टर संगीता कस्तूरे, डायरेक्टरेट हेड बायो मैन्युफैक्चरिंग को आमंत्रित करता हूं वेलकम रिमार्क्स के लिए. मैम, इट्स ओवर टू यू.
थैंक यू बालेंद्र. माननीय सचिव और डिपार्टमेंट ऑफ बायोटेक्नोलॉजी डॉक्टर राजेश एस गोखले, पद्म भूषण एंड पर्यावरणविद और हेस्को के संस्थापक डॉक्टर अनिल जोशी जी, इस वेबिनार में उपस्थित डिस्टिंग्विश्ड स्पीकर्स, डीबीटी के मेरे सहयोगी और ऑनलाइन जुड़े डीबीटी सहयोगी और अदर पार्टिसिपेंट, मैं आप सभी को इस वर्ल्ड एनवायरनमेंट दिवस, जागतिक पर्यावरण दिवस के अवसर पर जो वेबिनार आयोजित किया है उसके लिए आपका हार्दिक स्वागत करती हूं. विश्व पर्यावरण दिवस, जैसे बालेंद्र ने बताया, कि ये जागरूकता और कार्यवाही के लिए समर्पित सबसे बड़ा वैश्विक मंच है. इसमें पर्यावरण कार्यक्रम के जागरूकता के अलावा ये जैसे बताया कि 1973 से ये साल मनाए जाने वाले इस दिवस पर दुनिया भर के सारी सरकारी, उद्योग, वैज्ञानिक समुदाय और संस्थान और नागरिक सभी और इसकी तारीखी पर्यावरण के जुड़ी गंभीर चुनौती का समाधान किया जा सके और सस्टेनेबल डेवलपमेंट को बढ़ावा दिया जा सके, इसलिए हम ये सेलिब्रेट करते हैं. पर्यावरण का जागरूकता बढ़ाने के अलावा जो नए नए समाधान या सॉल्यूशंस बने उसको कैसे प्रदर्शित करें, इकोसिस्टम की रक्षा कैसे करें, बायोडाइवर्सिटी कंजर्वेशन और सस्टेनेबल डेवलपमेंट की दिशा में सामूहिक प्रयास जुटाने के लिए एक महत्वपूर्ण मंच दिया गया है. तो इसी के लिए आप सभी का सहयोग भी जरूरत है और हम साथ मिलकर काम कर सकते हैं. आप सभी ने इसमें जुड़ा, इसलिए सभी का मैं फिर से एक बार धन्यवाद देती हूँ. इस वर्ष का थीम है जलवायु कार्यवाही, जिसे क्लाइमेट एक्शन, जो यूएनईपी के वैश्विक अभियान दैट इज नाउ फॉर क्लाइमेट से जुड़ता है. इस अभियान का उद्देश्य दुनिया भर के समुदाय को जलवायु परिवर्तन के समाधानों को लागू करने और उनके लिए आवाज उठाने हेतु प्रेरित करता है. यह दिवस क्लाइमेट चेंज के निपटने के लिए सामूहिक कार्रवाई तथा तत्काल आवश्यकता पर जोर देता है और बढ़ते तापमान, लाइक एक्सट्रीम वेदर्स, आज भी हम देख रहे हैं दिल्ली में बारिश की हो रही है जिसकी वजह से हमें नेटवर्क इश्यू हुआ है, बायोडाइवर्सिटी कंजर्वेशन तथा क्लीन एनर्जी ट्रांजिशन, ऐसे मुद्दों को रेखांकित करता है. Recently, नीति आयोग has published a report, net zero scenario, in which from waste, almost in 2020, GHG emission from waste is almost 2,959 million tons of equivalent, while per capita emission is 2.2 CO2 equivalent. And since you all know that waste generation is increasing, hence the emissions is also growing, and we are expecting that this may grow from 75.64 million tons CO2 equivalent from 2020 to almost 266.110 million CO2 equivalent by 2070. So this would be almost 3.5 times increase in emissions. That is what it is projected by Niti Aayog. But this is mitigation is one thing which is the opportunity for all of us to how to utilize this waste, how to utilize this waste carbon and give value add to this, and biotechnology plays a very important role in this case. like biomethanation, biogas, CNG production, which can be used, plus methane recovery. These are so from almost decade, as mentioned by Dr. Balendra, we have been supporting bioenergy program, environment biotechnology, forest biotechnology towards sustainable development, climate change mitigation. All these multiple projects have been supported. Almost we have got 200 patents in this plus 450 publications in various areas. And we have also developed solutions in various areas. And today some of those would be showcased by the speakers in this webinar. I would also suggest that under the new initiative under BioE3 Biomanufacturing, we are also promoting various themes under this which are well aligned with the sustainable development and the climate change address goals. This is what I would like to hear, but I would like to hear from all of us and most excited to hear from डॉक्टर अनिल जोशी क्योंकि वो एक बहुत ही अच्छे पर्यावरणवादी है और उनसे सुनने के लिए हम सारे उत्सुक हैं। तो मैं अभी अपना यहीं पे समाप्त करती हूँ मेरे शब्द, लेकिन इसमें हमें सब जोड़कर मिलके हमें काम करने की जरूरत है और ग्रीन हाउस एमिशंस इंडिया के जो नेट जीरो पाथ है उसमें हम बायोटेक्नोलॉजी का कैसे सहभाग होगा तो उसकी अपेक्षा कर देते हैं और उसमें जो भी डीबीटी से सहयोग होना है दैट वी विल बी हैप्पी टू गिव दैट सपोर्ट once again on behalf of national environment world environment day on this eve of world environment day and this seminar I would once again welcome all of you to this webinar with this thank you all of you
बहुत बहुत धन्यवाद मैडम। आपने बहुत ही अच्छे तरीके से हमारा एनलाइटनमेंट किया जो विभाग की विभिन्न एक्टिविटीज हैं बायो3 पॉलिसी के अंतर्गत वो किस प्रकार जलवायु परिवर्तन को मिटिगेट करने में सार्थक सिद्ध हो रही हैं और डिपार्टमेंट ने विभिन्न जो अचीवमेंट्स दिए हैं अन्य परियोजनाओं के द्वारा। इसी के साथ हमारे सबके बीच में आज के मुख्य अतिथि इस प्रोग्राम के डॉक्टर अनिल प्रकाश जोशी जो कि पर्यावरणविद हैं जैसा कि मैडम ने बताया और आप सभी जानते होंगे एंड ग्रीन एक्टिविस्ट हैं और मुख्य रूप से उनको हैस्को जो कि हिमालयन एनवायरनमेंट स्टडीज एंड कंजर्वेशन ऑर्गेनाइजेशन है देहरादून उत्तराखंड में उसके फाउंडर हैं उसके संस्थापक हैं और हैस्को अपने आप में एक संस्था नहीं ये एक तरीके का जो एनवायरनमेंट से संबंधित विचारधारा है उसको आगे लेके जाने वाला संस्थान है। आदरणीय कोटद्वार में अवतरित हुए थे 6 अप्रैल 1955 को उत्तराखंड के रहने वाले हैं और निश्चित तौर पर वनस्पति विज्ञान और आपकी पारिस्थितिकी और एनवायरमेंटल साइंस के विभिन्न डिग्रियां और इस क्षेत्र में डिग्री से ज़्यादा इनका काम है जो कि एक रिवर जो पूरी तरह सूख चुकी थी उसका रिस्टोरेशन पूरी तरह से रिवाइवल जो बहुत बड़ा काम है। तो उनका जो वर्क है वो एक्सटेंसिवली सस्टेनेबल डेवलपमेंट या रूरल डेवलपमेंट और उससे संबंधित जो क्रियाएं हैं उनसे बहुत संबंधित रहा है हिमालयन क्षेत्र में और अन्य सभी क्षेत्रों में। इनको माउंटेन मैन के नाम से भी जाना जाता है क्योंकि कम्युनिटी बेस्ड जो प्रोग्राम्स हैं उनको इम्प्लीमेंट किया है फॉर रूरल लिवलीहुड जनरेशन। आदरणीय पद्मश्री और पद्म भूषण से सम्मानित हुए हैं। उसके अलावा जो मैं एक व्यक्तिगत रूप से जानता हूं क्योंकि उत्तराखंड में मैंने कुछ प्रोग्राम अटेंड किए हैं तो उनको वहां पे लोग सन ऑफ हिमालय के नाम से जानते हैं सन ऑफ हिमालय। सो इसके साथ ही सर मैं आपको इनवाइट करता हूं आपके वक्तव्य के लिए। स्वागत है सर, इट्स ओवर टू यू।
आवाज पहुंच रही है आपका? मेरी आ रही है?
आ रही है सर, नमस्कार। आइए सर, आ रही है आवाज सर।
हिमालय में मुझे का मौका मिल रहा है और
सर क्लियर नहीं आ रही आवाज आपकी, बीच में टूट रही है।
We cannot hear you.
सर हम आवाज नहीं सुन पा रहे सर आपकी। हम
डॉक्टर अनिल प्रकाश जी, क्या आप हमें सुन पा रहे हैं? क्या बाकी
अभी मेरी आवाज आ रही है?
हाँ, अब आ रही है
हाँ, अब आ
आपकी
रही है
आवाज।
आपकी आवाज। तो देखिए, पहली बात तो अब मेरे की आशा है कि मेरी आवाज बड़ी साफ आपके पास है, आशा करता हूं। और मैं हिमालय से हूं, इसलिए हिमालय की ओर से आप सबको प्रणाम। लेकिन कुछ बड़े मुद्दे हैं। इससे पहले मैं प्रकृति, पर्यावरण और आपके डिपार्टमेंट ऑफ बायोटेक्नोलॉजी का जो क्लाइमेट कनेक्ट है उसके बारे में मैं बात करूं। मुझे आपके सामने कुछ बात कहनी है, क्योंकि एक बात हम सबको कतई नहीं भूलनी चाहिए, और बिल्कुल भी नहीं भूलनी चाहिए, कि हम में से कौन है, वो कोई देश हो या व्यक्ति हो या किसी भी पोजीशन में हो दुनिया में, हर एक को पानी चाहिए, हर एक को प्राणवायु चाहिए, भोजन चाहिए करने को। और जब तीनों चारों चीजें हों तो इसका विभाग से लेना देना नहीं होता, इसमें हर व्यक्ति का लेना देना होता है। इसलिए कम से कम अब ये इश्यू जो है प्रकृति, पर्यावरण का, क्लाइमेट का, ये अब ऐसा इश्यू है जिसमें हर एक को गंभीरता से उसी रूप में सोचना, देखना होगा जिस रूप में कहीं सरकारों की को टटोलते हैं कि उनकी कितनी चिंताएं हैं। सवाल यह है कि ये सबकी चिंता का सवाल है और इसे बड़ी गंभीरता से लेने की आवश्यकता है। अभी अभी आंकड़े अमिता जी बता रही थी, तो आप ये देखिए कि दुनिया का कोई भी कोना अब ऐसा नहीं बचा कि जहां प्रकृति के किसी ना किसी रूप में हमें दंश ना झेलने पड़ रहे हो। अजीब सी बात है ये। और एक बात और सबसे बड़ी ये भी है कि अगर 1973 में 72 में स्टॉकहोम में एक बैठक हुई थी, बड़ी चिंता उठाई गई थी पर्यावरण को लेके, प्रकृति को लेके, और ये भी तय किया गया, चलिए आइए मिलकर के हम लोग एक पर्यावरण दिवस मनाते हैं जून 5। हम सब लोगों ने सामूहिक रूप से उसको स्वीकारा था। आप सब लोग इसको मानेंगे। आज कितने साल हो गए? 27 साल तब के और 26 साल अब के, 5 डेकेड्स। 5 डेकेड पहले एक मैंने सबने अंतर्राष्ट्रीय स्तर पर एक आवाज बनाई थी, एक इश्यू खड़ा किया था कि हवा, मिट्टी, जंगल, पानी और जो भी सो कॉल्ड डेवलपमेंट वी आर ऑल आफ्टर टू, उसके कारण हमको जो कॉस्ट देनी पड़ रही है वो इन्हीं रूप में देनी। बड़ी रोमांटिक अप्रोच थी स्टॉकहोम की कि इन्वायरमेंटल कंजर्वेशन होना चाहिए एंड सो एंड एवर सिंस द टू थिंग्स केम आफ्टर because you are all project guys also. No project now you get receive and comment where there is no word sustainability is used. पता है किस में सस्टेनेबिलिटी को यूज नहीं किया जाता। सारे प्रोजेक्ट में स्टेट प्लान हो, नेशनल प्लान हो, ग्लोबल प्लानिंग हो, there is one often word that we have been using, sustainability, that came in 1972 of course, ये तो आप जानते हैं। Now my simple thing which all of you really need and seriously consider, what is sustainable, and we were all what after. Look at नदियां जो हमको पानी देती हैं, पोखर, तालाब, ग्लेशियर एक एक करके खत्म हो गए। ये सब 50 सालों में हुआ होगा। इस सालों में जिसमें हर 5 जून को बैठ के आपने मैंने पर्यावरण दिवस को मनाया होगा और दुनिया में भी मनाया गया होगा, इस ग्लेशियर पानी के जितने भी वाटर बॉडीज हैं आइदर पॉल्यूटेड हो गए या मात्रा घट गई। एक छोटा सा आंकड़ा है, यू मस्ट ऑल बी नोइंग इट, कि 1950 में अराउंड वी हैड अवेलेबिलिटी ऑफ वाटर 5000 क्यूबिक लीटर एवरी वन, आज घट के 1300-1400 में चला गया। वाटर अवेलेबिलिटी इंडिविजुअल की बहुत ज्यादा कम हो गई। और मैंने क्या कहना है, मैं तो एक ऐसी जगह बैठा हूं जो हिमालय है, जो सारे देश की सेवा की है, हवा, मिट्टी, जंगल, पानी में, उस पर भी आक्रमण हो गया। उसमें भी कई तरह के आक्रमण है और आक्रमण हिमालय के लोगों के कारण नहीं। अनफॉर्चुनेटली द सेवियर आर सफरर। अब बताइए कि अगर दुनिया में ग्लोबल वार्मिंग, क्लाइमेट चेंज है, फॉरगेट अबाउट द लैंड, कम टू 2/3 पार्ट ऑफ द वर्ल्ड, अर्थ का सी, ओशियन किस तरह से तप रहे हैं, यू नेवर नो। एंड वेयर इज द मानसून, डेफिनेशन बदल गई। विच सीजन यू आर। फरवरी इतना गर्म हो जाता है कि अप्रैल है, मार्च ठंडा हो जाता है, फिर हमें कंबल ओढ़ने पड़ते हैं, और मई इतना गर्म होता है कि वो जून के मध्य की तपिश दे देता है। कहां हैं हम, और कौन सा किस तरह से सोचा जाए कि ये क्या, इट्स नॉट नेचर। नेचर ने तो 2 बार दुनिया को ढाया था। पृथ्वी में प्रकृति ने 25 करोड़ साल पहले, 6.5 करोड़ साल सब निस्तनाबूद कर दिया था। ऑल लाइफ टर्मिनेटेड दैट टाइम। एंड अब की बार जब लाइफ टर्मिनेट होंगी वो हमारी होंगी, क्योंकि हमने 10,000 साल पहले जो कुछ सोचा, सो कॉल्ड सिविलाइजेशन, पहला डिफॉरेस्टेशन किया, फार्मिंग लाए, और फिर फार्मिंग के लिए सेटलमेंट लाए, और सेटलमेंट के बाद सोचना बंद कर दिया, जहां जाएं जहां देखें जो चाहें लपेटे। हमने जानवरों को मार दिया, अन्य जीवों को लाइफ खत्म कर दी, और आज आप और मैं एक सिविलाइज वर्ल्ड कहलाते हैं और शहर ज्यादा। तो अब आप कैसे सिविलाइज हैं हम लोग, कि जरा सोचना चाहिए कि 53 years प्लस इन्वायरमेंट डे वी सेलिब्रेट एवरीवेयर इन द वर्ल्ड, बट अनफॉर्चुनेटली हमको उसकी मीनिंग आज तक ठीक ठीक पता नहीं है। पानी गया, हवा तो आप दिल्ली में रहते हैं, आपको मुझसे ज्यादा पता होगा व्हाट इज द AQI। 7 million प्रीमेच्योर डेथ आर रिपोर्टेड प्राणवायु के कारण, जिसे हम प्राणवायु कहते हैं। प्राण की भी चिंता नहीं है, वो तो हमने वो भी डहा दी। सॉयल, यू मस्ट बी नोइंग मोर देन मी, मिट्टी तो गई, माटी कहे कुम्हार से तू क्या रोंदे मोए, एक दिन ऐसा आएगा मैं रोंदूंगी तोए। आप जानते हैं, आई रिमेंबर 2005-6 की बात है कि पार्लियामेंट में उस समय जो मिनिस्टर होती थी, वो बड़ा, उन्होंने एक बात बोली जो बड़ा हिम्मत से नहीं कह पाई, कि पंजाब से एक ट्रेन चलती है जिसका नाम कैंसर ट्रेन है। क्यों नहीं बोला उन्होंने कि वो क्या बात थी, क्योंकि वी हैव यूज्ड सो मच ऑफ केमिकल फर्टिलाइजर, दैट्स वन। एंड एज आई से, मैं आई ट्रैवल होल कंट्री बाय साइकिल। हमने ये भी है कि केमिकल फर्टिलाइजर को जो प्रमोट किया है वो हजारदस हो रहा है, मेरे से ज्यादा आप साइंटिस्ट लोग जानते हैं, लेकिन रिपोर्ट्स भयानक किस्म की हैं। तो पानी गया, हवा गई, मिट्टी गई, और जंगल की हालात तो यह है जिसको हम एक तरह का मैन्युफैक्चरर मानते हैं, हवा, मिट्टी, जंगल, पानी का। आपके देश में 0.08 हेक्टेयर प्रति व्यक्ति अवेलेबल है, वो भी वो कि वो आंकड़ा जो फॉरेस्ट डिपार्टमेंट देता होगा, दावा करता होगा, और दुनिया में 0.5 हेक्टेयर है, क्योंकि दुनिया में आधे से ज्यादा जंगल समाप्त हो गए। अब मुझे बड़ा विषय मेरी इसको कभी समझ में नहीं आता कि हम ये जो दिवस मना रहे हैं, जल दिवस, पानी दिवस, जल दिवस, अब प्राणवायु दिवस भी आने वाला होगा बहुत जल्दी, उसके बाद वन दिवस मनाते हैं, ये दिवस मना रहे हैं, इसमें शायद एक नए सिरे से आत्ममंथन की आवश्यकता है। और ये विज्ञानी जितना होगा, वैज्ञानिक सोच जितनी होगी, आई थिंक हम इसके बेहतर तरीके के सॉल्यूशन निकाल पाएंगे। ये तो आपने सुन ही लिया कि दुनिया का सबसे व्हाट इज द क्रक्स ऑफ द प्रॉब्लम, दैट कार्बन इमिशन दैट वी हैव। अभी आप पता 37 से 40 बिलियन टन आपका प्रति वर्ष प्राण वो वायु में, एटमॉस्फेयर में ये जा रहा है, कार्बन जा रहा है। और आप ये भी जानते हैं कि उससे तो 28-30 गुना ज्यादा हानिकारक मीथेन है, और वो मीथेन आपका करीब 10 से 12 बिलियन टन मैट्रिक टन जा रहा है। और अभी तो फ्लोरोनेटेड कार्बन, उसका भी 1-2 बिलियन टन जा रहा है। जब इतनी ढेर सारी चीजें हैं आप और मैं उगलेंगे, डू यू थिंक दिस इज ऑल ह्यूमन इंड्यूस्ड? हमने किया है ये, इसके लिए कोई दूसरे प्लेनेट नहीं है। अब कितनी अजीब सी बात लगती है, वी कैन नॉट डिनाई दैट वी शुड नो व्हाट इज देयर इन द मून एंड द मार्स। हमको हमारी क्यूरोसिटी होनी चाहिए, साइंस की क्यूरोसिटी होनी चाहिए। उसमें ढूंढ क्या रहे हैं हम? उसमें पानी ही ढूंढ रहे हैं। अब कितनी अजीब बात है, मंगल और चंद्रमा में पानी ढूंढो, और अपनी दुनिया में जो पानी है वो खत्म हो रहा है, खराब हो रहा है, उसकी चिंता उतनी बड़ी नहीं है। अब आप देखिए सॉल्यूशन, साइंस सॉल्यूशन क्या है, वेयर आई समटाइम मुझे बड़ा खराब लगता है, साइंस सॉल्यूशन फॉर गंदे पानी का क्या निकल के आया, या पीने के पानी की स्कैर्सिटी आई, क्या सॉल्यूशन आया? वाटर मोटर बॉटल बिजनेस आ गया, ठीक पानी उपलब्ध कराएंगे आपको, और वो एक बहुत बड़ा बिजनेस बन गया, बिलियंस डॉलर। फिर वाटर प्यूरिफायर आ गए, हर घर को चाहिए वाटर प्यूरिफायर, वो साइंस सॉल्यूशन है। दिस इज व्हाट वी आर इनटू। एंड मोर इंपॉर्टेंट इज कि हमने सॉल्यूशन भी जो निकाले, ऑल्टरनेटिव भी जो निकाले, वो ज्यादा भयानक है। मैं आपको रिपोर्ट बताता हूं कि जो हम इन जो इकोलॉजिकल क्राइसिस है, इकोलॉजिकल क्राइसिस के जो सॉल्यूशन ऑल्टरनेटिव निकाले जा रहे हैं, वो और ऐड करेंगे प्रॉब्लम्स को। ये बात फैक्ट है, बट अनफॉर्चुनेटली नो बॉडी इज देयर टू फाइंड आउट कि इसको भी एस्टिमेट कर लिया जाए कि हमारे जो साइंटिफिक जो हम ऑल्टरनेटिव निकाल रहे हैं उसका क्या होगा। अब हम फंस गए। सच बात यह है कि वन ऑफ द स्टडी सेज, आई बिलीव दैट द आईपीसीसी सेज इट इज हार्डली 10% चांसेस ऑफ रिटर्न। तो भाई आप सच तो बात यह है, प्रकृति को नहीं बचाना है, अपनी अपनी जान बचानी है। पहले तो ये फॉर्मूला बड़ा तय होना चाहिए, वो स्टॉकहोम की बैठक हो या उसके बाद की जो फॉलोअप है, दुनिया भर में तमाम बहस है। एक हर साल कॉन्फ्रेंस ऑफ पार्टीज होता है, आप जानते हैं, कॉप जिसे कहते हैं। हम सब जो लोग उतावले होते हैं वहां जाने के लिए, समटाइम आई फील इसका तो इट हैज टू बी रिकॉइंड, इसको बोला जाना चाहिए कंफ्यूजन ऑफ पार्टीज, क्योंकि हमको पता ही नहीं है कि हमको जाना कहां है। यू कैन नॉट यू कांट स्टॉप डेवलपमेंट एंड नोबडी वांट टू डू दैट। एवरी कंट्री छोटा होगा, वो अपने शेयर का डेवलपमेंट चाहिए उसे। वो कहे व्हाई शुड ही नॉट यूज द फ्यूल, तो फॉसिल फ्यूल वो क्यों नहीं यूज करेगा? वो कहेगा मैं उसके बराबर जाना चाहिए, मैं भी अमेरिका नहीं बन सकता तो अमेरिका से छोटा हूं। तो हम लोग कहीं बहुत कंफ्यूज हो चुके हैं, बहुत बढ़ चुके हैं। तो इसलिए शायद अभी जो सबसे बड़ा चैलेंज जो मुझे लगता है कि कुछ चीजों को आप रोक नहीं सकते। अब आप कुल मिला के देखिए, आपके डिफरेंट बड़े सेक्टर जो है, आपका मैन्युफैक्चर से 30% कार्बन एमिशन है, ट्रांसपोर्टेशन 16 है, इलेक्ट्रिसिटी जो हमको चाहिए जो कोल से ज्यादा आती है अपने 35% है, एग्रीकल्चर भी 12%, उसमें कम नहीं, वो भी अपना है, और वेस्ट और अदर जो है। अब देखिए कितनी अजीब बात है, एक तरफ हिमालय टूट रहा है, एक तरफ हिमालय का पहाड़ ढह रहा है, क्योंकि हिमालय में जो तमाम वो आ रहे हैं, डिजास्टर आ रहे हैं, इट इज नॉट बिकॉज ऑफ द हिमालयन पीपल, बिकॉज ऑफ द ग्लोबल वार्मिंग विच अनफॉर्चुनेटली अदर्स आर डूइंग, कॉजिंग बाय अदर्स। तो जो समुद्र तप गया, उससे जो बारिश होगी वो हमारी सर पे आके गिरती है और हमको डहा देती है। और आप तो रिपोर्ट पढ़ते हैं हर मानसून कितनी भयावह है। आपको स्वीकार कर लेना चाहिए कि बरसात किसी समय पे हम लोग बहुत वेलकम करते थे, बरसात का स्वागत करते थे। आज हम डरे रहते हैं जब पानी पड़ता है। ये स्थिति पहुंच गई। और किसके पहाड़ टूट टूट के नीचे आ रहे हैं। और अब एक नए पहाड़ है जो आपके दिल्ली में भी 3-4 पड़े हुए हैं, प्लास्टिक के पहाड़। अब वही पहाड़ों में घूमिए, उन्हीं पहाड़ों में देखिए। तो मुझे लगता है कि जो हमारे शहर और गांव जो बटे, तो हमने सब कुछ नॉलेज हब माना शहरों को, लेकिन जो विजडम थी हवा मिट्टी जंगल पानी की, वो गांव से चलती थी, वो नॉलेज गांव का होता था, उसको हमने इग्नोर कर दिया। और इसीलिए इंडियन नॉलेज सिस्टम को लेकर जो हमने शुरुआत की थी एचआरडी के साथ, इट वाज वी बिहाइंड। तो हमने कहा था कि यही होना चाहिए, जो हमारी कई चीजें नए विज्ञान की अपनी आवश्यकता मना नहीं की जा सकती, लेकिन पुराने विज्ञान को, परंपराओं को नकारा नहीं जा सकता। अब आप इन सबको भी अगर छोड़ दें, आपको क्या लगता है किसी सरकार में क्या अब इतना दमखम बचा है कि इसको अंडू कर दे? को हो ही नहीं सकता। कि 8 बिलियन पीपल आर कंज्यूमर, और मान के चलिए कि आप कोई भी रास्ते निकाल लें, अगर आप कंज्यूमर हैं तो आपके कार्बन फुटप्रिंट इंडिविजुअल भी हैं, कलेक्टिव भी हैं, बड़े बड़े कार्बन फुटप्रिंट हैं। और उन कार्बन फुटप्रिंट को भी छोड़ दें तो हम लेट अस कम टू द अनदर। इसी लुक एट द मार्केट, लुक एट द मार्बर, कितना चमकीला मार्केट बाजार हो गया। कितने तरह के तो चिप्स ही चिप्स हैं, और क्या टेढ़ा मेढ़ा जो भी तमाम की चीज है, या जो तमाम तरह के प्रोडक्ट बाजार में बेच रहे हैं जो हमको अट्रैक्ट करते हैं, यूथ को खास तौर से। हम में से किसको पता है उसका इंडिविजुअल कार्बन फुटप्रिंट क्या है, या हमने कितना शेयर किया? And that I was 2 days back in Delhi. We had a very serious concern about carbon footprint. हमने इसमें कहा we today only sending a letter to the Prime Minister. हमने एक प्रार्थना की है, आप जब कोई प्रोडक्ट बनाते हैं, आप भी डीबीटी के कई प्रोडक्ट हैं, उस प्रोडक्ट में आप लिखते हैं कि साहब इसकी कितनी कैलोरी है, इसका एक्सपायरी क्या है, इसके इंग्रेडिएंट क्या है, तो भाई ये भी लिख दो इसमें कितने कार्बन फुटप्रिंट है। क्योंकि आप एक जीन्स जो हम पहनते हैं उसके 4 से 5 किलो कार्बन फुटप्रिंट है 1 किलो पे। वेफर खाते हैं उसके 2 से 3 किलो कार्बन फुटप्रिंट है। जैम, जेली, स्क्वाश जो भी हम बाजारों में वो चमकीली चीज जो बिक रही है, वो पिज्जा जो नया भोजन हमारे बीच में पर आक्रमण कर चुका, उसके 2 से 3 किलो कार्बन फुटप्रिंट है। और ये जो तमाम चीजें केएफसी और ये मैकडोनाल्ड, ये जो बड़े बड़े स्टोर आज हमारे बीच में आ गए फैशन बतौर, वो पता नहीं खाने को कैसा है, पर फैशन बतौर तो यूथ वहीं जाता है, सबके हाई कार्बन फुटप्रिंट है। तो हमने एक प्रार्थना की है, हम प्रार्थना कर रहे हैं प्रधानमंत्री से कि आप अब ये भी कहिए, इन जो कुछ भी मैन्युफैक्चरर हैं, बड़े बड़े घराने हैं जो प्रोडक्ट को बनाते हैं, वो इस पे ये भी लिख दें भाई इस बारे आटू चिप्स में या इसमें तोड़ मरोड़ जाने क्या क्या नाम है, मुझे ज्यादा पता भी नहीं है। मैं जब भी किसी दुकान को आप चले जाइए, कितनी रंग बिरंगी वो दुकान हो चुकी, लेकिन उसके पीछे का कालापन हमको पता नहीं है, कितने कार्बन फुटप्रिंट है उन सबके। ये सवाल बहुत बड़े हैं। और मेरा सोचना व्यक्तिगत ये है कि वैसे तो जो मैं सोच रहा हूं बड़ा गलत है, इस जीवन में तो संभव ही नहीं है कि उसके बारे में सोचा जा सके, लेकिन ये हमारी पीड़ाएं हैं। हम जो इन मुद्दों पे काम कर रहे हैं, तो अभी हमें एक ही बात लगने लगी। मैं आपको सत्य बता रहा हूं अपने हृदय से, जब लगातार पिछले 40 सालों से इन मुद्दों में दाएं बाएं टेक्नोलॉजी, नॉलेज, साइंस हम जो करते थे, हमने एक बात तो पाई है कि प्रधानमंत्री का एक नारा बड़ा सटीक है, लोकल फॉर वोकल। और आप में से बहुत से लोगों को पता है, हेस्को ने 1992 में एक नारा दिया था, लोकल नीड मीट लोकली। उसका कारण ही था, जितना होममेड प्रोडक्ट होगा उसके कार्बन फुटप्रिंट उतने होंगे, जितना बाजार से उसी प्रोडक्ट को खरीद के फ्रेंच फ्राइज खाएंगे आप, उसके कितने होंगे, लेकिन घर में बना दोगे आलू की चीजें तो भाई उसके कुछ और होंगे। तो वो लोकल फॉर वोकल में शायद एक ये बात भी हिडन फैक्ट है कि कार्बन फुटप्रिंट जितने लोकली होगा उतना कम होगा, और जितनी इंटेंसिव प्रोसेसिंग होगी उसके कार्बन फुटप्रिंट ज्यादा होंगे। ये तो आप सब लोग इस बात को गंभीरता से स्वीकारेंगे। अब सवाल ये पैदा होता है, मैं तो कभी कभी हम लोग अपने को अकेला पाते हैं जब हम साइंटिस्ट उनको सबको इस तरह से जोड़ के, क्योंकि एक बहुत बड़ी बात है जिसके लिए मैं बहुत सी आईआईटी से जुड़ा हूं। वी हैव आईआईटी कानपुर विद अस, एमयू ट्राइन, आईआईटी रुड़की, उनकी क्या बीओजी में भी हूं, और आईआईटी भागलपुर, आईआईटी पटना। मेरा उनसे एक ही प्रार्थना है, दिस इज द हाई टाइम फॉर रेलेवेंट रिसर्चेस। यू शुड नॉट मेयरली डू ए रिसर्च फॉर ए प्रोजेक्ट एंड ए रिपोर्ट वाइंड अप और भेज दी। इट हैज टू बी वेरी रेलेवेंट। और मेरे आप सबसे प्रार्थना है दोनों तरफ के लोगों से, रिसर्च की रेलेवेंस इज वेरी इंपॉर्टेंट। और क्योंकि अगर हम ये नहीं करेंगे, अब आप देखिए जितने पीएचडी वीएचडी हो रहे हैं, हेस्को में भी 15-20 रिसर्च स्कॉलर हैं जो कि दे आर सपोर्टेड बाय इंडिविजुअल आईआईटीज एंड द इंस्टीट्यूट्स, उनको डिग्री वहां से मिले, लेकिन रेलेवेंट रिसर्च पे काम करेंगे हमारे साथ। ये एक मुद्दा हमने खड़ा किया है नया। लेकिन मुझे लगता है कि एक बड़ी बात जो आप लोगों की ओर से मैं कहना चाह रहा हूं, मैं गो डीपली इनटू द बायोटेक्नोलॉजी और उसके प्रोडक्ट, उनके इश्यूज पे, तो मैं आई फाइंड इट कि यस, बायो मैन्युफैक्चरिंग विच यू हैव बीन यूजिंग इन ए क्लाइमेट इशू दैट द वे यू आर ट्राइंग टू एड्रेस, मेरा आपसे एक प्रार्थना है, हाउ मच कार्बन फुट यू वर एबल टू रिड्यूस by your different product, is that there for the public consumption? I think you must not have calculated, but that's a very important issue, क्योंकि आप बहुत बड़ा एथेनॉल की बात की है आपने, आपकी तमाम मेडिसिन आती हैं, मेडिसिन आ रही हैं, और नए नए प्रोडक्ट जिसमें आपका टूल जो है वो बायो है, और बायो टूल और वो प्रोसेसिंग भी उतनी इंटेंस नहीं है, कुछ को छोड़ के, लेकिन उनका कार्बन फुटप्रिंट कितना है इंडिविजुअल का, that's a very important. Now how would you compare you with the other institutes? That by saying that आपके रपट होनी चाहिए कि in a given year, अब वही क्यों देना, हमने कितना पैसा खर्च कर दिया और कितना बचा हुआ है, ये भी बता दीजिए कि आपने कितना कार्बन फुटप्रिंट अपने प्रोजेक्ट के माध्यम से, अब प्रोडक्ट के माध्यम से आपने रिड्यूस कर दिया। तो उसकी एक पिक्चर जब सामने होगी, लोग इस पर विश्वास करेंगे। और सच्ची बात तो ये है, एक केमिकल जो अंग्रेजी दवाई टाइप की चीज होती है, और उसमें आप में बहुत अंतर होंगे, बहुत से प्रोडक्ट हैं, ऐसा नहीं है कि बहुत ढेर सारे प्रोडक्ट हैं। और मेरा मानना सबसे बड़ा सेक्टर एक एग्रीकल्चर भी है। यू नो, वी आर थर्ड लार्जेस्ट कंज्यूमर ऑफ फर्टिलाइजर, प्रोड्यूसर भी है और कंज्यूमर भी है। तो अब आप कल्पना कर लीजिए कि हम कार्बन फुटप्रिंट तो जो कर ही रहे होंगे, लेकिन जो फुटप्रिंट उसके पड़ेंगे आप और मेरे भोजन पर, वो और बड़ा सवाल है जिसको कि हमको कर देता, आपकी रिस्पॉन्सिबिलिटी सडनली बहुत बढ़ जाती है। और जब अगर हम इस तरह की साइंस की सेटिंग करेंगे, नो व्हाट इज अदरवाइज, आई ऑलवेज हैव बीन सेइंग, मेरे को आपको पता है मुझे एफएनए मिला था इस बार, और मुझे एफएनए जब मुझे मिला तो मैं वहां गया भाषण देने के लिए, तो मैंने सभी साइंटिस्ट लोगों से हाथ जोड़ के यही बोला था। अब पता नहीं इट वाज प्लीजिंग और उसने क्या असर दिखाया होगा मुझे नहीं पता, लेकिन मैंने उस बात को चूका नहीं ये कहने के लिए कि रेलेवेंस ऑफ रिसर्च इज द बिगेस्ट चैलेंज। हम किस तरफ जा रहे हैं, हमारे पूरे जर्नल भरे हुए हैं, तमाम है, पता नहीं क्या क्या रिसर्च इस देश दुनिया में हो चुकी होगी, लेकिन आज बड़ा समय इस बात का है कि हम करें। और बायोटेक्नोलॉजी में जो भी कोई मुझे सुन रहे होंगे, आपके कोई नीति धारक होंगे, जो भी होंगे, या वो जो वन हु विल बी लाइकली टू बी द पार्टनर एज ए प्रिंसिपल इन्वेस्टिगेटर विद यू, तो मेरा आपसे एक बहुत बड़ी प्रार्थना ये है, आपकी बहुत सी कोशिशें हैं, लेकिन कल मिला जुला के जब आप एक गंभीर बैठक बायो मैन्युफैक्चरिंग और ये कर सकते हैं, तो ये बात भी करिए कि क्लाइमेटिकली द वे यू आर कंट्रीब्यूटिंग, व्हाट इज द स्केल, नंबर वन, व्हाट इज योर प्रिंट, उसमें आपके अपने क्या फुटप्रिंट है, कार्बन फुटप्रिंट आपने कितना रिड्यूस किया, ये सब सवाल। वरना आप ये मान के चलिए कि 1973 के बाद एक और पर्यावरण दिवस आया, एक और बैठक आपने कर ली, और अगली बार जब आएंगे तो फिर तब पता नहीं कितने कार्बन फुटप्रिंट और हो जाएंगे। और अब दशा इतनी खराब होनी चाहिए कि आप ये मान के चलिए कि हमने दुनिया को ट्राई टू रूल द वर्ल्ड बट द रूइंड इट। सच्ची बात तो ये है, हमने रूल करना चाहा है होमो सैपियंस इस स्पीसीज ने। तो ये होमो सैपियंस स्पीसीज जब से आई थी, उसने पहले तो क्या किया, जितने भी होमो की स्पीसीज थी उसने होमो नेंडरथाल मारे, इरेक्टस मारे, रघुरास्ट्रिस, सबको किया एक एक की बात किनारे, और ओनली स्पीसीज दैट केम कॉल्ड द होमो सैपियंस। तो जब उसने बाकी जानवर वानवर मार दिए, तो अब अपने आप को मार रहे हैं। ये तमाम युद्ध देश दुनिया में हो रहे हैं, ये क्या है, किस बात की लड़ाई? आपको पानी, आपको भोजन है, आपका सब कुछ है, पर आप वेपन्स के पीछे ज्यादा है। किसके लिए? कोई शेर बाग थोड़ा मारना है, मिसाइल चाहिए, एक दूसरे को मारना है। हम वो हैं, और जब हम ऐसे हैं और अगर नहीं समझे हैं तो फिर अंत की तैयारी करिए। प्रकृति का कुछ नहीं बिगड़ेगा, प्रकृति बदल जाएगी, दूसरी चीज आ जाएगी। पहले भी 6.5 करोड़ साल पहले, 25 करोड़ साल पहले ऐसे तो डायनासोर गए, कई थे, सब चले गए, हम आए, हम भी चले जाएंगे। तो बस सवाल यही है कि हम अगर बुद्धिजीवी कहलाते हैं और हैं तो उसका सही परिचय दें। और सही परिचय इस बात में छुपा है कि अगर हम साइंटिस्ट हैं, हमको इस चीज को देखना चाहिए, समझना चाहिए, विज्ञान के उस पहलू को जो प्रकृति के विज्ञान में बहुत कहता हूं, मैंने कहा साइंस ऑफ नेचर को लेके कुछ काम ही नहीं है। वी डू नॉट नो, एंड आई इनवाइट ऑल ऑफ यू हियर टू माय प्लेस वेयर आई वुड शो यू व्हाट इज द साइंस ऑफ नेचर। नदी सूख गई थी, अभी जिक्र हुआ, हमने कैसे उसको पानी लाया, नेचर ऑफ साइंस ऑफ नेचर। अब हम बहुत हाई क्लास साइंटिस्ट नहीं हैं, पर लेकिन हमें मुझे लगता है प्रकृति के विज्ञान के पक्षधर हैं। और इसी नदी को देख के अभी आजकल बड़ी चर्चा है वाया नदी बिहार की, वो भी वापस आ गई। और ऐसे कई नदियां जन्म लेने लगी हैं। 2009-10 का ये केस था। मेरा मानना है कि जहां हम हाई साइंस को पढ़ें तो हम उस साइंस को भी जानें जिसने आज तक हमारा साथ नहीं छोड़ा, हमने छोड़ा है। और जब हमने छोड़ा है तो फिर आप समझिए कि प्रकृति भी आपका साथ छोड़ देगी, क्योंकि उसने आपको जिंदा रखा है। मैं ज़्यादा इसलिए नहीं जोर लगाना चाहता बोलने में। मुझे आशा है कि मेरे बोलने का जो कुछ भी होना होगा वो तो तथाकथित है ही है, लेकिन मेरी प्रार्थना यही है कि आपके साथ जो तमाम लोग जुड़े हैं वो शायद इन मुद्दों को अगर ध्यान में रखें और औपचारिकता न निभाते हुए हम में से व्यक्तिगत रूप से भी, सामूहिक रूप से भी, अपने प्रोग्राम्स में भी अगर इस इशू को गंभीरता से लेंगे तो शायद हम कुछ असर डाल पाएंगे। वरना तो ये भी एक दिन और है और ये भी गया, मेरा भी चला गया। आप सबको प्रणाम और धन्यवाद।
बहुत बहुत धन्यवाद
बहुत बहुत
डॉक्टर
धन्यवाद
और
डॉक्टर अनिल। और
कैन यू हियर अस डॉक्टर श्रीश?
कैन यू हियर अस डॉक्टर श्रीश?
यस आई कैन हियर यू नाउ।
यस आई कैन हियर यू नाउ।
तो बहुत बहुत धन्यवाद सर। मुझे कुछ पंक्तियां याद आ रही हैं इसी पे जितना आपने बोला उस पे कि सूखती नदियां, दरकते पहाड़, बदलती जलवायु, कम होती पृथ्वी की आयु, ये नुकसान सबका है। प्रयास बढ़ाएं, अनुशासन अपनाएं, यही तो काम हम सबका है। बहुत बहुत धन्यवाद सर। निश्चित तौर पे हम ये जो इनसाइट्स आपने शेयर की हैं इनको कहीं ना कहीं प्रोग्राम में किसी न किसी वर्टिकल में, ये निश्चित तौर पे प्रोग्राम एक्सपेंशन में और उसको आगे बढ़ाने में काम करेंगी, कहीं ना कहीं इंक्लूड होंगी परियोजनाओं के रास्ते, किसी न किसी रास्ते। हम सब ये कार्बन फुटप्रिंट जो एक मेजरमेंट आपने बताया है, जो इंडिविजुअल बेसिस पे भी मेजर किया जा सकता है कि हमारा क्लाइमेट चेंज में क्या रोल है और हम उसको कैसे मिटिगेट कर सकते हैं, ये बहुत आवश्यक है और ये हर किसी की रिस्पॉन्सिबिलिटी है। इस जिम्मेदारी को भी हम करेंगे कि अपने जीवन में उतारें। सर एक प्रश्न तो कई सारे हैं ऑनलाइन, लेकिन एक प्रश्न ये है, पता नहीं ये बहुत डिटेल में जाएगा, मेरा यही निवेदन रहेगा कि आप थोड़ा सा ब्रीफ में टाइम के इंटरेस्ट में बताएं। प्रश्न ये है सर कि जो आपका रिजुवेनेशन का था, जो हैस्को जो रिवर है उसको कौन सा ऐसा टेक्नोलॉजी आपने प्रयोग किया था जिससे आप इसको टाइमली अचीव करने में सक्षम हुए? ये एक ऑनलाइन प्रश्न है सर। आपका माइक शायद म्यूट।
सर अनम्यूट करिए। हाँ
बहुत आपका धन्यवाद। आपने बड़ा सही सटीक प्रश्न मुझसे पूछा। देखिए ऐसा है, इस बात को आप सब जानते हैं कि प्रकृति का सबसे बड़ा प्रबंधन है, उससे ऊपर हम कभी जा भी नहीं सकते। कि प्रकृति के प्रबंधन में पानी, जंगल, हवा, मिट्टी हमको उपलब्ध कराई गई। उसी कड़ी में पानी लाने का रास्ता हमेशा प्रकृति से, जो हमारे ज्यादा नदियां देश में, हमारे देश में खास तौर से रेन फेड है। ग्लेशियर नदियां तो गिन के हैं, लेकिन ये बड़ी नदियां तभी बनती हैं जब रेन फेड रिवर से मिलती है। और रेन फेड रिवर का जो बड़ा माध्यम होता है वो उसके कैचमेंट होते हैं, जो वाटरशेड भी कहते हैं। उनमें लगे हुए जो जंगल होते हैं वो उनका माध्यम होता है। और जंगलों का पानी, जंगल में पानी पड़ता है, फिर वो अपने कैपिलरी मूवमेंट से ये नीचे उसमें चला जाता है, रूट से नीचे चला जाता है, और वो एक्विफर को रिचार्ज करते हैं, और एक्विफर ही रिवर को रिचार्ज करती है, स्प्रिंग को रिचार्ज करती हैं। इतना हमें पता था, पर हमें यह भी पता था कि अगर आज पेड़ लगाएंगे तो 12 साल से पहले तो पेड़ पैदा नहीं होगा। तो हमने बड़ा सिंपल काम किया, हमने 1 हेक्टेयर में 300 वाटर होल बना दिए, ट्रेंचेस, वाटर होल हमने उनको कहा। और उसका सबसे बड़ा फायदा ये हुआ कि जो पानी गिरे, मतलब 30 मिलीमीटर रेनफॉल में, वो 1/1 क्यूबिक मीटर का जो वाटर होल है वो भर जाता है, और एक साथ उसमें आपका 10,000 लीटर पानी चला जाता है। मतलब 1 हेक्टेयर में 300, तो आप कल्पना कर लीजिए कि कितना लीटर पानी उसमें गया होगा। और हमने 44 हेक्टेयर को ट्रीट किया। हमने फॉरेस्ट डिपार्टमेंट के बड़े हाथ जोड़े, उन्होंने कहा यार जंगल में गड्ढा खोदेंगे, क्या करवा रहे हो डॉक्टर साहब। अरे मैंने कहा भाई मैं नहीं करवा रहा हूं, प्रकृति कह रही है, कर दीजिए। देखिए आश्चर्य क्या हुआ, नदी में दूसरे साल तो पानी आया, ये कोई बड़ी बात नहीं थी, लेकिन आप और मेरे लिए जो सबसे बड़ा लेसन था कि जब पानी आता है तो सब कुछ आता है। आपके शरीर में, मेरे शरीर में, पेड़ में, पृथ्वी में सबसे ज्यादा पानी है, पानी के उत्पाद हैं हम। तो जानते हैं जब वहां पानी आ गया तो छोटे छोटे पौधे आ गई, छोटे छोटे पौधे आए तो खरगोश आ गया, खरगोश के बाद पोर्कुपाइन आ गया, फिर उसके बाद छोटे छोटों के बाद फिर हिरन आ गए, और हिरन के बाद लेपर्ड आ गया, और लेपर्ड के साथ साथ फिर यहां से 12-15 बर्ड स्पीसीज थी, 115 बर्ड स्पीसीज हैं हमारे यहां। आप जो भी अद्भुत 4-5 तरह के किंगफिशर हैं, हॉर्नबिल्स 3 तरह के आ गए हैं, और अनगिनत नंबर डव आ गए। देखिए हम पानी लाना चाहते थे, तो प्रकृति के विज्ञान का ये असर है, हम पानी लाना चाहते थे, बाकी साथी अपना आप आ गए। सरकार ने बड़ी ज्वलित हो गई इसको देख के, और इसे नेचर पार्क अनाउंस कर दिया। अब आप हैस्को में आएंगे तो एक गेट लगा होगा, उसमें लिखा होगा नेचर पार्क, क्योंकि वहां अब, और यहाँ पे दून स्कूल से लेके सारे बच्चे, दून स्कूल की पहली क्लास यहीं लगती है। हम उन बच्चों को साइंस ऑफ नेचर सिखाना चाहते हैं, और इस तरह से बहुत से लोग जुड़ गए। नमामि गंगे ने उसको उठा लिया और उसको प्रोग्राम को अलग ले जा रहे हैं। कई जगहों से अब उस नॉलेज को हमारे स्तर पे भी और अन्य स्तर से भी इसको देश में ले जाया जा रहा है। बड़ा सरल था, 1 हेक्टेयर में, एक 10 मीटर स्क्वायर में एक वाटर होल के पीछे कारण था कि उसके लिए कैचमेंट हो जाएगा 10 मीटर स्क्वायर, तो इसलिए हमने ये गड्ढे खोदे, और ये अब एक आंदोलन बन गया है देश में। अभी मेरे पास डायरेक्टर आए थे नेशनल वाटर मिशन के, ही वाज सो एक्साइटेड अबाउट इट, और इस तरह से लोग आते ही रहते हैं। आप लोग भी आइए, आपको प्रार्थना, आप लोगों से तो मैं लंबा समय जुड़ा रहा, अब आप लोगों ने छोड़ दिया या मैंने छोड़ दिया, ये तो बात की बहस और बात होगी, लेकिन मुझे लगता है कि इस तरह की टेक्नोलॉजी नॉलेज की आज बहुत आवश्यकता है। और आज हमारे बीच में जब आप आएंगे, देहरादून से और यहाँ के टेम्परेचर में 5-6 डिग्री सेंटीग्रेड का अंतर है। हमारी अपनी नदी है, अपना जंगल है, अपने जन जीवन और जानवर है, हमारा सब कुछ अपना है, और ये सुंदर जीवन है, बड़े ठाट में हैं हम यहाँ पे। आप भी आइए हमारे साथ, भात दाल खाइए, पढ़िए लिखिए, भागीदारी करिए।
4-5 साल पहले आई थी
4-5 साल पहले आई थी इसको, so it was very fortunate
to meet you and see the nature,
you and see the nature,
science of
the
nature,
science of
closely. आगे बढ़ो।
Yeah. So, thank you so much, sir, for this insightful, talk. Now it's time for the nodal ministry, and for this we invite Dr. R.V. Lal, senior scientist from Ministry of Environment, Forest and Climate Change. There was minor modification in agenda because of technical glitch, so that's why it is here. So we request you, sir, for your remarks for 5 minutes before secretary. Yeah. Thank you so much.
Okay. Good afternoon, respected dignitaries and esteemed guests, and especially thanks to Dr. Anil Prakash Joshi sir, giving the insight with respect to the various issues related to the climate change. And I thanks to the DVT for giving this opportunity to participate this important webinar on adopt biomanufacturing to address climate change on the occasion of Eve of the Bird Environment Day 2026. The theme of Bird Environment Day is basically, we all know, that is climate action, joining the UNEP global campaign Now for Climate to mobilize communities, worldwide to implement and advocate for climate change. As we know that it's basically the collective effort of various land ministries to handle this very difficult task. Accordingly, the Government of India is implementing several programs and schemes under the overarching framework on National Action Plan on Climate Change. And there are 9 missions basically, and focused on this solar energy, energy efficiency, water, sustainable agriculture, health, Himalayan ecosystem, sustainable habitat, Green India mission, and static knowledge for the climate change. And these whole and NFCC basically aligned with respect to the state action plan on the climate change. At present, 34 states and UTs in the country, they prepared their SFCC plan and implementing with respect to the climate actions. And as we are aware that India, our country is a party to the United Nations Framework Convention on the Climate Change, its Kyoto Protocol, and the Paris Agreement. Under the Paris Agreement, recently in April 2026, India has submitted its enhanced NDC for the period 2031 to 2035. And lot of due to the various effort by our land ministries and state government, our country has now enhanced its emission intensity reduction targets to 47% by 2035, and against the target related to the non-fossil fuel based installed solar electric power capacity, our country has already achieved 53% non-fossil fuel capacity as on this March 2026, thereby meeting the earlier target 5 years ahead of the schedule. And we know that our country has further enhanced its ambitions to achieve 60% share of non-fossil fuel based energy resource in the installed electric power capacity by 2035. And these are the NDC, basically first NDC we have given in 2015, and then updated in 2022, and recently this NDC updated in this April 2026. And as we know that it's a very challenging and very difficult task, but Government of India, with the help of other land ministry and the state government, leading by example on the global stage, proving that sustainable development is driven by an unique blend on ancient wisdom and modern innovation. Though the transformative vision of mission life, and that is the lifestyle for the environment, we are turning abstract habits to protect the earth. Under the urgent campaign called Now for Climate, we must move beyond awareness to mass public participation, whether by saying clean up, clean energy mission, expanding our electric mobility ecosystem in our country, or participating in the nationwide Ek Ped Maa Ke Naam tree plantation drive. Therefore, with the help of on behalf of the Ministry of Environment, Forest and Climate Change, this ministry appreciate the effort of the Department of Biotechnology for doing a lot of research in the similar field. Let us collectively take a pledge today to adopt an eco-friendly lifestyle, protect our natural heritage, and secure a cleaner, greener, and sustainable tomorrow for generation to come. Jai Hind, and thank you very much to all of you.
Sir, for this, sharing the views from the Ministry of Environment and Forest and Climate Change, that is the nodal ministry also. And it is interesting to listen about various activities. Obviously, the Department of Biotechnology and MOFCC, Department of Biotechnology activities under the biomanufacturing, under BioE3 policy, those are very much, complementing to many of the activities, even under mission line working on, water remediation. So we are complementing there. Also on biofuels, so energy part also we are complementing, like alternatives to single-use plastics, biopolymers, biochemicals. These are the things. Those it's like a converging approach where we are in together we are contributing towards whole of the government approach, and in this way we can contribute for the nation. मुझे 4 लाइन याद आती हैं कि कम उपयोग अधिक सहयोग, कम उपयोग अधिक सहयोग, रिसोर्स बचाएं, कुछ छोड़ते जाएं, आवाम सबका है. With this, thank you so much, sir. Now, this is time I will invite Dr. Rajesh S. Gokhale, Sir, Secretary, Department of Biotechnology, TG Brick and Chair BIRAC for his talk. It's over to you, sir. Can you make in?
Namaskar and a very good afternoon to everyone. It gives me a pleasure to join you all today in celebrating World Environmental Day 2026, a day of global significance and urgent reflection. The Department of Biotechnology is proud to mark this webinar showcasing DBT efforts in one of the most pressing challenges of our time, the climate change. Happy to note that Dr. Anil Prakash Joshi, a distinguished environmentalist and is the keynote speaker for this webinar, he has devoted decades to promoting community-led environmental conservation and sustainable resource management, and he has worked towards climate-resilient developments in the Himalayan region. We are happy to see him today here, and also he's learned from his experiences of how to do environmental stewardship and climate action. Immensely pleased is to find several distinguished investigators talking in different thematic domains and taking forward and championing effective scientific and technological solutions. World Environmental Day serves as an important reminder that environmental sustainability is not merely an ecological concern, but a development imperative. The theme of this year, climate action, and the global campaign now for climate emphasize the urgency to translating commitments into measurable actions. The Department of Biotechnology has aligned its programs to support India's climate and sustainability goals through research, innovation, demonstration and deployment projects of biotechnology-based solutions. This is an opportunity to showcase and share the innovative biotechnology-driven solutions addressing climate change. Under the BioE3 policy, the department is supporting high-performance biomanufacturing for sustainable fuels, chemicals, materials, and for various other resources which will create pathways for a circular and low-carbon economy. As is evident from the scientific discourse, the BioE3 policy has helped us integrating the bio-based innovations to industrial opportunities through dedicated infrastructure for scaling up the innovations, through biofoundries and biomanufacturing hub with thematic focus on important areas for the industrial growth, manufacturing like carbon capture utilization, bio-based chemicals, biopolymers, climate smart agriculture, etc. It is time now about building resilient communities, safeguarding public health, strengthening economies, and creating sustainable pathways for future generations. While the window to address climate change is narrowing, meaningful progress remains possible through collective actions. On this World Environmental Day, let us reaffirm our commitment to harness science and innovation for a sustainable future. Let us transform environmental challenges into opportunities of green growth, resilient ecosystem, and a prosperous bioeconomy leading towards a Swachh, Atmanirbhar, and Viksit Bharat. Thank you and namaskar to all of you.
Thank you so much, sir. We follow the directions you had given through this talk. This is very important, and we will follow all the things in our work program, and work program will be taken up further accordingly. With this, now we will be moving to the showcase talk from the lead DBT supported investigators with lines की सतत सुरक्षित रहे प्रकृति ये काम सबका है परिवेश बचाने वालों में नाम सबका है. So the first talk is by Dr. Praveen Kant, Indian Institute of Science, Bangalore. This is on delimiting the species boundary among Hanuman langurs using integrated taxonomy. So, sir, it's over to you. We request you to kindly finish it in 5 minutes.
Yeah, sure. Can everyone hear me?
Yes, we can hear you.
Okay, thank you. I'm trying to share my screen.
Make
How
him
the hell do
presenter. I think you can share the entire screen, sir.
Entire screen, okay. Oh God, what is it doing now?
Yes.
Is
Yes,
it better?
we can see your. Yes, it is good.
You can see the presentation, right?
Yes, sir. Please go ahead, sir.
Okay. Okay. So I
Request
talked
you to
about.
finish in 5 minutes. Thank you.
Yes, I'll just quickly talk about a DBT-funded project where we looked at Hanuman Langur diversity. And the main inspiration for this work was the huge amount of morphological diversity that we see among Hanuman Langurs. And in fact, if you look at the classification of Hanuman Langurs, it's quite a mess. There are like many different classification schemes, and each one of them has a different uh, taxonomy. Some call it one species with multiple subspecies. Others have classified it into multiple species and so on. So what we did was, in fact, you see these major tail carriage forms in North India and South India. So what we did to resolve this issue was to actually characterize the morphological diversity of Hanuman langurs across India, various characters, tail carriage, presence and absence of crest, and so on. Collected this data from across peninsular India. We also collected fecal samples to do the genetic work, and that is what was supported by DBT. And the morphological work basically showed that there are at least uh, 3 species of langurs, morphologically distinct forms in India. Of course, this did not include the Himalayan population. So basically, in North India, there was one morphological form called Entellus. And then in South India, there were 2 forms that people had called Hypoleucus and Priam. And then we went on to do some molecular work. This is a tree based on the mitochondrial data. And you can see on top you have Entellus, the northern form, which is found throughout plains of northern India. They all cluster together. They have a very distinct tail carriage. And then in South India, you have 2 forms, Hypoleucus and Priam. The tail loops away from the head. And also within South India, you have another species of langur called Nilgiri langur. So Nilgiri langur is completely different. It is also branching with Priam, right? So basically what people called Hanuman langur consists of at least 3 genetic clusters, Entellus, Hypoleucus and Priam. And morphologically too, they are forming 3 distinct clusters. So there's multiple lines of evidence suggesting that these are at least 3 different species, right? And then we looked at nuclear data. They also separate out the 3 species of Hanuman langurs, Entellus in North India, Hypoleucus and Priam in South India. When we look at the genetic data using a program called Structure, which basically tries to um, decipher the different genetic clusters within your genetic data. Once again, we find 3 clusters. Of course, here I've also included Nilgiri langur, so there are 4 clusters here. What about ecological or environmental data? So we did some niche modeling, um, and each of those species have distinct environmental niches. Entellus in northern India, Hypoleucus and Priam in South India, and then in the Western Ghats you have Jonai, which is Nilgiri langur. So environmental data, morphological data, and genetic data all support splitting Hanuman langur into at least 3 different species, one in northern India and 2 in South India, right? Now, what about the Himalayan langur? So this subsequently we extended this work to include Himalayan langurs. So these are langurs confined to the Himalayas. So we sampled from Kashmir to Sikkim. And once again, you have a distinct clade of Himalayan langurs, a distinct cluster, genetic cluster of Himalayan langurs that are distinct and different from the langurs in the plains. The langurs in the plains, the one called Entellus, found in northern plains. So these two are completely distinct, and we used the same approach, looked at morphological character and ecological character, and found the same result. So here's the overall synthesis. What we used to call as Hanuman langur, and we thought was a single species distributed from the Himalayas all the way to Sri Lanka, actually consists of 4 different species. Right, and we have ecological, morphological, and genetic evidence to support this. In the Himalayas, we have Semnopithecus cetaceus. In northern plains, all the way through central India and parts of Orissa, we have Semnopithecus entellus. In South India, we have Semnopithecus hypoleucus, and further south, we have Semnopithecus priam. So, thank you, everyone.
Thank you so much, sir, for fantastically, summarizing the outcomes of the research that you had taken on the taxonomy of different Hanuman langurs and how it is relevant and related to different places in India from north to south. And this was fantastic insights. And this is very much important aspect, from climate change viewpoint also, that how this taxonomy or this distribution is being affected with changing climate. So that could be another interesting area that we could think of. Thank you so much,
Thank
sir.
you.
Now we are moving to next. This is turning carbon into livelihoods. Algae powered biomanufacturing for green jobs and reduce synthetic nitrogen dependency. The speaker is Dr. Adi Viswanathan from Pinnacle Biosciences. So this is over to you, sir. Kindly finish it in 5 minutes.
Yeah,
Thank you so
I'll
much.
share.
Start sharing the result. In this screen, sir. Okay.
Good evening, respected advisor, Biomanufacturing Directorate, Secretary DBT, and distinguished guests, and fellow researchers and industrial leaders. Today, I would like to share how algae-based carbon capture and utilization can address the 3 major critical issues today India is facing. Traditionally facing, like first one is industrial carbon emission. Second thing is our the chemical fertilizer crisis that we are that is happening today. Third is employment generation. Traditionally, these 3 challenges can be addressed independently. We believe that algae biomanufacturing offers an opportunity to solve them together. So that is what today I wanted to present here. So the chemical fertilizer crisis, the global consumption of the chemical fertilizer today has been accounted to be 205 million metric tons, whereas 50% of them is nitrogen fertilizer. And coming to the average nitrogen use efficacy, the global standard, if you are seeing, it is only 55%. And approximately 50% of the nitrogen is being lost. It is not available to the plant. So coming to the consequences of these chemical fertilizers that we is happening today is, see, like 90% of our chemical fertilizer is being imported in India is being imported. We import approximately 15 million tons of chemical fertilizers annually. So which results in rising of import bills, high fertilizer subsidies, soil degradation, water pollution, nitrous oxide emissions, and supply chain vulnerability, as like the war times, which we are facing. Coming to the economic impact, India spends approximately 1.7 to 1.9 lakh crore annually on fertilizer subsidies. So we are looking for an alternate way where India's fertilizer challenge is no longer an agriculture issue. It is simultaneously on a food security, economic security, and climate change. So we propose algae-based solutions for sustainable agriculture, where these chemical fertilizers can be gradually replaced by these algae-based products. Why algae? See, these algae are primitive plants which can grow in areas where agriculture cannot happen. And any kind of water, like saline water, wastewaters, can be used to cultivate algae. And they are like 10x more faster when compared to that of land plants and forests in their growth. And it, as these algae are single cell protein, it is a whole nutrition where it has good protein, nutrient like minerals and sugars, so that when it is being used as fertilizer, the absorption of the nutrients to the plant is the availability is very high. And when it is used in soil, it enhances soil microbial activity. And also now, to have a net zero agriculture, these algae based fertilizers, mainly these blue green algae, which has this heterosis, which absorbs atmospheric nitrogen, can be used to have a carbon negative agriculture and so on. And apart from nutrition, these are all also acting as a very good biostimulants. So apart from nutrition, it also gives a very good biostimulant property to the plant. So it can be a very good replacement for synthetic fertilizers, and that as the crisis which we are happening today. The objective of our project is not to completely replace the synthetic fertilizer, but to produce more food with less imports. So coming to the carbon capture through algae biomanufacturing, so we'll have a small max. So if India substitutes 10% of its imported fertilizer, which will be approximately 1.5 million tons of chemical fertilizer into algae derived input, the sources of our the algae cultivation is our carbon dioxide, and the sources of carbon dioxide may be the thermal power plants, distilleries, cement plants, fermentation industries, or biogas plants. So to grow 1 kg of algae, approximately 1.3 to 1.8 kg of carbon dioxide is required. And 1 kg of algae will be approximately having 30 to 40% of protein. And when it is converted into nitrogen, it will be around 4 to 6% of nitrogen. So which is very similar to that of nanourea, which has 4 to 6% of nitrogen. And again, the highlight here is not only the nitrogen, because today the nitrogen availability, the absorption of nitrogen is a big question. So as algae are single cell protein, apart from nitrogen, this also has good amount of minerals and sugars, which will make this nitrogen absorbed by the plant. And here is a small max, like when we are substituting 1.5 million ton of fertilizer into an algae based product, we will be growing 1.5 million tons of algae, which will be internally sequestering 2.7 million tons of carbon dioxide. And for every ton of CO2, we can give one microbiologist or biotechnologist a job can be provided. So it means that approximately 2.7 million jobs can be provided just by sequestering 1.5 million tons of by producing 1.5 million tons of algae. And the oxygen generator is huge. It will be like 1.8 million tons of oxygen is being synthesis. So these are all the benefits when we are substituting algae as a carbon capture. So algae transformation carbon emissions for a liability into a biological feedstock. So we provide the industries and solution which generates a revenue to the industry, unlike other chemicals like amine technologies and all those things. So this is a process which we are doing for our algae production. So we have introduced a two unique proprietary system in this industry, which is a carbon dioxide infusion system. As CO2 is a poorly soluble gas, we have designed a system where CO2 is efficiently dissolved in water, and that water is used for algae cultivation. And we have a unique methodological high-raised algae culture system, which is like a competitor, or which will be much more better than the raceway tanks and the photobioreactor, which is being conventionally used in algae cultivation system. And this is our system which we are proposing. So this makes algae cultivation very feasible and economical in large scale CO2 sequestration and algae cultivation. And we already have a product which is made from microalgae protein digested into amino acid, and which is already in the market. So we are just going to like copy paste this in a large scale. So that is what we are trying to do, and we are with these industries we are discussing to make this algae CCS happen and to give lot of green jobs for our fellow Indians. So, for concluding statement, the future of sustainable agriculture will not be defined by how much nitrogen we apply, but by how efficiently we use it. And the future of climate action will not be defined by how much carbon we intake, but how the carbon is being valued, how the value chain of the carbon is happening. That is what we are working into. Thank you, Jai Hind. And thanks for providing this opportunity on this Global Environmental Day. Thank you.
Thank you very much, sir. This is fantastic approach to convert or make available the sustainable carbon for the agriculture purposes, or you can say repurposing of the waste carbon for benefit of the agriculture and reducing import dependency, as well as towards Atmanirbhar Bharat while fighting with the climate change too. Thank you so much for being with us. So now we are moving to serial number 3. This is closing the carbon loop, transforming CO2 from biomethanation into value. Here the speaker is Dr. Manju D. Tanwar from Organic Recycling Systems Limited. So it's over to you, madam. Kindly finish it in 5 minutes. Thank you so much.
Thank you. Thank you so much. Can you see the screen, sir?
You can share.
I
But
have shared.
we can't see right now. We are unable to see. Yes, I think go ahead. Yes. Now we can see, just make it full screen mode and go ahead. Yes,
Now,
ma'am, please go ahead.
yeah.
Perfect, perfect.
Yeah. Good afternoon, everyone. Today, I would like to thanks to all because DBT BioRec, I'm participating this event today. So I'm really thankful to entire DBT and BioRec team also, and all available dignitaries present here. So my topic is closing the carbon loop, and it's connected to the BioCCUS project also, which has been awarded from DBT BioRec. So under this, actually, first I would like to introduce a little bit about my organization. Organic Recycling Systems Limited has established in strong presence in bioenergy sector. So particularly in biomethanation process, where organic waste and agriculture waste is converted into valuable resources, specifically bio-CVC and biogas through electricity. So under this project, we have proposed, because biogas contains carbon dioxide and methane, both almost in similar amount. So CVC is utilized as a replacing fossil fuels, but carbon dioxide unutilized. Still, people are searching about the better technology to utilize this CO2, but it's still not utilizing properly. They are trying various technology for converting this CO2 into value-added product. Under this project, we have proposed two approaches. One is biological approach, that is biological fixation through algae, and the second one is photocatalytic approaches, where CO2 is converted into mixed alcohol through photocatalysis. So this is the project. I'm not taking much time for this. So basically, the theme of this project is, we are not focusing on the CPG, we are focusing on CO2 only. So after separation, CO2 can be utilized and diverted to two reactor streams. One is photocatalytic reactor, which is converted through photocatalysis and visible light, presence of the visible light. And the other approach is biological fixation, because algae cannot utilize entire CO2, because if we will consider 100 ton of organic fractions of MSW plant, so it's around 8 to 9% CO2 generated every day per day. So it cannot be utilized entirely through algae. So that's why we have used photocatalytic approach also here. So maximum we can utilize this CO2 into converting into value added product through photocatalytic way. So here I have given few benefits what happen if we utilize this. So instead of waste, CO2 become a secondary feedstock of value addition of the biomethanation plants. So because of that reason, up to 20 to 30% increase overall carbon utilization efficiency increases, economical benefit and environmental benefit also through this particular technology. So this is actually my project. I only take 5 minutes only. Thank you so much, sir. Thank you. This is my project. Thank you.
Thank you so much, madam, for looping the carbon emissions that is happening out of the bioenergy process and presenting this bioenergy integrated carbon capture, utilization and storage thing. This is very much important for looping this carbon and making the biofuel production processes or bioenergy processes more cleaner as compared to today. So thank you so much. Now, actually, there is some request. Somebody is from outside, so we are taking number 10. This is dry times at forest ages, seedling traits and response to drought. Dr. Meghna Krishnadas from National Center for Biological Sciences, Bangalore, would be presenting here. So it's over to you, Dr. Meghna Krishnadas. Can you hear us? So I think
Dr. Meghna, are you there?
There is some connectivity issue, so we can continue with the number 4. This is, can the oldest bacteria help make biomanufacturing sustainable on Earth and space? So Dr. Sri Srivastava from International Center for Genetic Engineering and Biotechnology, New Delhi, is the speaker here. So it's over to you, Dr. Sri. Thank you so much. I think we request you to finish it in 5 minutes only.
Yeah. Can you see my screen?
Please go ahead. Perfect.
Yeah. Good afternoon, everyone. And first and foremost, thank you to Dr. Sangeeta Kasture and Dr. Bale for giving opportunity to present our research, which is on can we repurpose the oldest bacteria to help biomanufacturing sustainable on Earth and space. So now Dr. Adi Vishwanath has already set up the problem for me, and we are also trying to address the same problem that our country and the world is currently facing, the carbon and the nitrogen challenge in terms of sustainability. We are emitting too much of carbon dioxide, leading to global warming. India is the 3rd largest emitter of CO2. And on the nitrogen challenge, actually, we are also a 3rd largest importer of urea in the world, which is provided to the farmers so that they can grow their crops to feed the population of humans as well as cattle. Interestingly, a lot of nitrogen is excreted as urea in urine. And also, very interestingly, actually, instead of being an importer, we can in fact become a urea exporter if we can recycle the urea from urine. When we talk about bioprocesses, the same is there, that if we can recycle the carbon and nitrogen, we can improve the sustainability of our bioprocesses as well. So, and not only on Earth, even on ISS, the International Space Station as well, there is an efficient system of recycling of water, but there is still no efficient method for recycling carbon and nitrogen. And this is one of the major challenges in designing long-term human stays for space. So with this background, I want to briefly introduce cyanobacteria, which are the oldest and fastest growing phototrophs. These were actually the first photosynthetic organism with fossils dating more than 3 billion years old. And at that time, Earth was very rich in CO2, Earth's atmosphere, and these cells grew under those conditions. The long evolution has also helped them to become really, really robust and also very efficient. So unicellular strains are the fastest growing phototrophs. There are also many marine strains which we are focusing, and they would not need fresh water. And being photosynthetic, they can utilize carbon dioxide, and we can also make them utilize urea to produce oxygen and a nitrogen and antioxidant rich biomass, which can be used as a fertilizer, food, and feed. The challenges on using cyanobacteria for carbon capture and recycling with the real industrial kind of waste is that the flue gases have very high CO2 concentration as well as sulfur dioxide and nitrogen dioxide, which is basically toxic to these cells. And the same is the problem with urea in urine, which is at very high concentrations. So this would actually leads to bleaching of cell as shown here. So what we have done is we have used a combination of adaptive lab evolution as well as process optimization to ensure that these cells can tolerate high CO2 and high urea concentrations. So here is where we are showing one setup where we have utilized an indigenous marine cyanobacterium to capture carbon from a simulated flue gas. This cyanobacterium was adapted so that it can now tolerate high CO2 concentrations, and we have used a very high CO2 concentration of 18% in the flue gas, simulated flue gas, and with a single pass we were able to capture over 96% of the CO2 using this indigenous marine cyanobacterium. We are also very fortunate to be a part of the Axiom 4 mission last year, which was part of the DBT's BioE3 policy, where our experiment cyanobacteria on ISS was conducted aboard ISS, and ISRO and ESA were also involved in executing this experiment aboard ISS, where we compared the growth of two cyanobacterial strains on urea or nitrate under microgravity conditions. We included an Indian isolate of spirulina and a very fast-growing Senecococcus strain in our study for their obvious beneficial properties. Because manuscript is in preparation for submission, I will not go into the details of the results of this, but give you a snapshot of what we have done. This is how our hardware looked, which contained the two different types of cells. And this is astronaut Shubhanshu Shukla conducting our experiment aboard the ISS. Not only this experiment, we have continued our work in this area, and now we are able to grow cells under much greater urea concentrations, approximately 10 times higher than what we had done before. So with that, I'd like to thank the Department of Biotechnology for the support that it has provided, ICGB Director, Faculty and Administration for the support, NFMC of Bharatidasan University for providing native cyanobacterial cultures, and also lot of my students who have worked in this area and have made this successful, as well as my collaborators. So our overall approach can be summarized in this slide. We are aiming to usher in an era of photosynthetic biomanufacturing where CO2 and urine are not waste but a resource. Thank you.
For this integrated approach, so you have connected biomanufacturing with all three, Jal, Thal or Nav. It's like this. So this is very much integrated. Thank you very much for sharing the insights. So now we are moving to next. This is serial number 5. So here speaker is Dr. Krishna Ray from West Bengal State University. This is restoring biodiversity in Sundarban's mangrove niches, success with indigenous plant growth promoting rhizobacteria. So it's over to you, ma'am. We will request you to kindly finish in 5 minutes. I think you can share entire screen and then just move to your slides. Yes.
I cannot find it. Which one is this?
No, I think. No. You just open the presentation.
Yes, I have opened it. Then?
And then go to the share button. Share is there, na?
Share screen.
In bottom panel.
Huh. I'm going there.
Just go to
then
share.
It's showing your screen.
Yes. Entire screen.
Yes, now it has come. Okay.
Yes, perfect. Go ahead.
Okay.
Yes, we can see. Perfect.
Thank you, DBT and BioRec team, for giving me this opportunity to talk here. This is about restoring biodiversity in Sundarbans mangrove niches, success with indigenous plant growth promoting rhizobacteria. Under the DBT support, since 2014, we have been working on a mangrove biorestoration project in Indian Sundarbans. And under this project, we have so far we have restored around 65 hectares non-protected degraded mangrove niches, and we have converted these niches into which were earlier degraded, from where the mangrove species were already almost lost. On those sites, we have converted them to multi-species, self-sustaining dense forest ecosystem, which are resembling just like mini Sundarbans. And each year, thousands and thousands seedlings, they are coming there, being recruited naturally, and they are making the sites more denser each season, and thus we can see that the ecosystem got repaired, which were earlier damaged. So not only that, under this project, we were successful in reintroducing, rather we can say reintroducing, several rare and threatened species which are already lost from the shorelines of these non-protected shorelines of Indian Sundarbans, like Heritaria fomes. This is endangered species. This Sundarban was the name after the name of this species, but now we cannot find them actually in the western part of Sundarbans. From the shorelines, it's totally lost. But we have been successful in creating germplasms of this germplasm of this Sundari now in several of our sites. Not only Sundari, Phoenix paludosa, this is also a near threatened line, brown Loya tursa, Insia ajuga, these are all under threatened category, and some locally rare species also, Nipa fruticans, Meropangulata, Xylocarpus, Lumnizeda racemosa, and many others. We have now our sites have become the source of germplasm for these individual species. But what we have seen that all these species, they are not showing uniform growth in all sites of restoration. So some sites are very much challenging, and where they're just surviving and their growth is becoming stunted. So what to improve the growth, what we have added in our restoration experiments, we have added one component by application of indigenous plant growth promoting rhizobacteria. Over the last decades, with DBT support, we have been able to isolate almost 400 bacterial isolates from the rhizospheres of Sundarban mangrove niches, and we have screened them in the lab under standard experiments, their plant growth promoting activities. And from that, we have started screening these PGP rhizobacteria, and we have started applying them to the degraded mangrove sites. So here today, I am just going to tell you about one experiment where we have taken two consortium. One consortium was entirely made up of actinobacterial isolates. We actually wanted to see whether these actinobacteria solely it is able to improve the growth of the mangrove or not. So this, and then we made another consortium. This is consisting of non-actinobacteria, mainly proteobacteria, gamma proteobacteria, this firmicutes. They were included under these, and we have screened all of them under laboratory conditions. Many of them are good nitrogen producer, nitrogen fixers. We have tested them by ARA assay, all these things. So what happened? We actually made large-scale cultures, and we have immobilized the cultures in biochar using the sodium alginate, and we made the beads. We took them to our sites, and we applied them. This is how we applied them on our sites, and you can see even after 28 days of so we could trace those biochar beads which are holding the bacteria, because there is a chance of washing out in Sundarbans. If you apply the bacterial culture, there is always a chance of washing out. So for that, we always immobilize this biochar, this bacteria on these biochar beads. This is this was totally these protocols were invented by our group of students, and they have also invented one indigenous machine for putting it in large cultures, putting it in large amount, because we need a large number of these biochar beads, bacteria immobilized beads for application. So I'm telling you about one experiment. This started in only May 2024, and this is a December 24 picture. You can say from here we have started. Here at this time they are same. They are the applied bacteria applied sites and without bacteria applied sites, they are almost same. But now this is a very recent picture on 12th April. You can see how the bacteria non-bacterial applied sites, these control sites, their growth is much less as compared to the sites where we have applied the actinobacterial consortium and also the non-actinobacterial consortium. You can see this is on the experiment was done on Phoenix paludosum, another near threatened line. These are totally on-site experiments with 75 plants each under each category, and you can see that this actinobacterial application after actinobacterial application plants are very palms are basically a very slow growing species, but here you can see how they are moving to the forming the pinnate leaves, and this is also almost after 2 years we could achieve this thing. And this is the above ground scenario. You can see always the actinobacterial application is doing better performance, significantly better than the other control and non-actinobacterial things. But what is happening at the below ground? What is the bacterial communities changes occurring? So for that we have done next generation sequencing, and here
sorry to intervene, ma'am. We request you to kindly yes
you can
speed
see
up and wind up.
we have seen a very interesting observation where we have applied actinobacteria. There the indigenous actinobacterial population got decreased, but the Firmicutes population, it got increased. And where non-actinobacterial population we have added, there we have seen that actinobacterial population got increased. So this is how we think this is the mechanism how they are going to improve the bacterial this plant growth, and we are doing further experiments on this, and we are going to metatranscriptomics now to exactly showing what are the PGPR genes and from which bacteria they are going to contribute there as a for improvement of these plants. And we have particularly found that one actinobacteria is having higher population in non-actinobacterial sites, and another Firmicutes is having higher population in the non-actinobacterial in the actinobacterial sites. So this is a very interesting observation. We are still continuing with these experiments. So these are our field staff who are supported by DBT and my students and the collaborators. I thank you for DBT for supporting us since long for doing this work. Thank you.
Thank you so much, ma'am, for showcasing this fantastic work. And Sundarban is one of the biggest carbon sink of the world, and really you are contributing in those harsh conditions. This is very much important activities. How it can be taken up, we understand it very much. It is very important initiative. Activities being taken up by you under DBT supported initiative. Thanks a lot for sharing these insights. Now we are at serial number 6. This is biochemocatalytic transformation of biorenewable resources into green transportation and sustainable aviation fuels, from Professor Azaz Ahmed, Indian Institute of Technology, Indian School of Mines, Dhanbad, IIT ISM Dhanbad. So it's over to you, sir. We will request you to kindly finish it in 5 minutes.
Thank you, sir. Thank you so much. Am I audible?
Perfectly audible.
Yeah.
Please go ahead.
So,
We
sir,
can see your slides. Perfect.
Yeah, thank you, sir. So here I'll be discussing briefly about the biochemocatalytic transformation. But before I start, I would like to thank the DBT and the committee members for inviting me here. Most of the problems that I prepared has already been discussed, so I will skip them. What are the major problems? One is definitely the solid waste. The other is the greenhouse gas emissions. And as Dr. Joshi rightly highlighted, there is one more thing that we are missing is the methane emission, which contributes roughly to 16% of the total greenhouse gas emissions. So in order to I can say, in order to tackle these problems, we have found another way that instead of thinking the agro residue or the kitchen waste or the other things as a waste material, can we consider it as a resource material? And if you see here, almost everything that we can make from a fossil fuel, like chemicals, energy, fertilizers, or anything that we are making at present, can be prepared from the agro residue as well as the kitchen waste, etc. In particular, if I talk about uh, we have solid fuel, we can make bio oil, syngas, power, biochar, or we can make high value chemicals that includes the aviation fuel, green gasoline. And in this regard, we got a project from the DBT that was particularly on the integrating the biological and the chemical aspect of engineering together to make the aviation fuel. And the reason is obvious. Approximately 10 kg of CO2 is emitted per second when a commercial aeroplane flies. So herein, IIT Delhi, IIT ISM Dhanbad and IIT Dehradun came together to develop a process that basically synthesizes the biomolecules via the fermentation route, and then we apply the butyric acid. We use a chemical catalytic route to convert that into the aviation fuel. Now, after converting that into aviation fuel, we also test it whether it is suitable or not. And what we found that here, this up to 93% conversion can be achieved when we are trying to convert this biomolecule into a suitable aviation fuel, that is called butyl butyrate. And this butyl butyrate can be blended up to 50% without changing anything in the existing aviation engine. So that actually helps to reduce the CO2 emissions or the greenhouse gas emissions 2% contribution of aviation industry to 1% straight away if we use the biomass molecule derived fuels. Herein, like we already have several patents and publication coming out from it. But motivated from this, what we did, we thought that this butyl butyrate is made from butyric acid and butanol. So can we make similarly for the gasoline and diesel? So we took up a project where we make the alcohol and levulinic acid together. And what we do, we develop a product called ethyl levulinate, which is a replacement of the gasoline. And this molecule, the ethyl levulinate, actually replaces the MTBE and polyaromatics. And for that, we also thought that there is a lot of requirement for chemical catalysis. So is there any way that we can find that what is the most suitable descriptor that actually helps to decide a catalyst without doing a lot of experiment? And we found that deprotonation energy, that means the energy required to take out one hydrogen from the catalyst, is responsible for the activity. And we tested it here for a lot of alcohols like ethanol, propanol, butanol, and we had two catalysts. One was the phosphotungstic acid, phospholinic acid. And what we found that the difference between the phosphotungstic acid and phospholinic acid was exactly 18.6 kilojoule, which is equal to the difference in their deprotonation energy. So this is activation energy, this is deprotonation energy. This indicates that if I know the deprotonation energy of a catalyst, I can easily find out whether this catalyst will be active or not. The second thing, when I'm changing it to ethanol, it becomes green gasoline. If I'm changing to butanol, it becomes green diesel. If I change some other reactant, it becomes aviation fuel. So there also we can find out whether this catalyst will be active or not. And for that, what is needed, if you see here, by increasing the carbon number, ethanol, propanol to butanol, so we are going from C3 to C4. The activation energy is increasing in both the cases. But the overall delta, the difference between the activation energy remains same. So this was one of the observations. Motivated from this, because we did the aviation fuel, we did the gasoline, we also moved to green diesel. And what we did, we changed the molecule, just this ethanol by butanol. And we were successfully able to identify that we can make the green diesel, and that all these molecules have been synthesized below 100 degrees Celsius, which is in contrast to the existing processes which operates around 800 degrees Celsius or 700 degrees Celsius and uses hydrogen. Herein also what we found that the activity of the catalyst is directly correlated with the acidity as well as the kind of addenda atom and the central atoms we are having. Besides that, if my number of protons, like here you see 4 hydrogen, 5, 6, here 3, and then addenda atom is changing. So the catalyst activity without doing any experiment in actual can be calculated theoretically, and we can synthesize a wide range of new catalysts. And that all will be helpful. Either we make green diesel, we make green gasoline, or we make sustainable aviation fuel. So this is one part that we have done on the transportation fuel. Similarly, the another major emitter of the CO2 is the steel industry. So there what we are doing, we are converting this biomass into biocoal. And this biocoal has potential to straight away reduce the CO2 emissions by 40% in the steel industry. Similarly, there is one more thing that Dr. Joshi earlier highlighted during his plenary talk, that we are not talking about the methane, this part. So IIT ISM Dhanbad, we are in the coal mine region, and coal mines has lot of coal bed methane. And coal bed methane, as of now, is left, and it's a greenhouse gas emission. So it causes at least 25 times more CO2 emission. So what we are doing, we have taken this coal mine methane, I'll skip the data part, and we are converting this coal mine methane into the carbon nanotube and hydrogen. And carbon nanotube sells around 5 lakhs rupees per kg. So the technically or theoretically, I can say the hydrogen becomes free of cost. These are few of the carbon nanotubes that we have made. This is another example, because methane is the smallest molecule. We went for plastic also, which is the largest hydrocarbon molecule. And we are able to make here, I can show that we are able to make the hydrogen very successfully. So this is all. We are trying to align our research with the different initiative of the government of India and the vision of the Honorable Prime Minister. And these are my funding agencies. Of course, DBT has been very instrumental in initiating this transportation fuel and aviation fuel research. And this is my group. We got last year awarded also by the Petroleum Minister for CO2 emission reduction. And this is our HCBM car. These are my collaborators. Thank you. Thank you so much.
Thank you so much, sir. This is good, and we can think of how can connect the intermediates of SAF, and this is one of the burning issue in recent times, and current situations always lead to innovate toward and scale up towards production of sustainable aviation fuels or any kind of intermediates as well as other biofuels. So obviously this is very futuristic approach, and world is looking towards the future fuels everywhere. So whatever towards future fuels we can contribute, we have to contribute. The thing is how we can integrate this approach with the other leads, those has been generated till butanol or bio-based butanol, we can say. So that we can think of further. Thank you so much for enlightening us.
Thank you, sir. Thank you so much. Thank you, ma'am.
So now we are going to serial number 7. Here the speaker is Professor Puneslow K. Bahaduri for Indian Institute of Science, Education and Research, Kolkata. So this is how can microbiomes drive sustainability and bioeconomy, learnings from the field. So it's over to you, sir. Then we will take the presentation of Dr. Meghna. Please go ahead, sir.
Thank you, sir. Can you see my screen? Is it visible?
No, sir. No. You have to share the entire screen. Just go to share, then share entire screen and open the slides.
Okay, sir.
You had not shared yet anything.
Okay, sir. Yes, sir. Now, sir?
Perfect, sir. Perfect. Please go ahead.
Thank you very much, sir. Thank you, Dr. Kasturi ma'am and Dr. Balendra Singh for this opportunity. I'll very quickly show how the indigenous microbes can be used to address critical questions linked to sustainability and also drive the bioeconomy of our country. The first example I'll give you is here here is an example where we have done community microbiome engineering, and top left hand side you can see we have deployed these community microbes without any genetic engineering, of course, put them in the inlet systems of two big sewage treatment plants. One is 8.3 MLD and one is 6.5 MLD in West Bengal. And what we have shown is that these intelligent microbes, they can remove the fecal coliform in 30 days. Essentially, there is continuous flow of it, and the fecal coliforms are getting removed, the organic pollutants are removed in the inlet water of the STPs, and it keeps on holding for 1 year. So no more deployment of the bacteria is required. And the net advantage of this approach is that the STPs can perform much better, essentially. The second example I'm showing you, bottom left hand side, is many of the semi-urban areas, they have the septic tanks. It is very common in eastern part of India. So the septic tanks are now being connected to the 16th FC through the fecal STP program. So what we have done is we have put again bunch of intelligent microbes, and they are able to eat up all the sludge, everything pretty much, including sanitary pads. And not only that, they hold and remove the fecal coliform bacteria from these septic tanks. Essentially, the water that comes out, the grey water, is now suitable for application in any field, for example, recreational forestry, cleaning the streets, or can be again linked to water purification system for further reuse. I quickly move from this knowledge of the application of these microbes in STPs and septic tanks. What we have done over the last couple of years, we have done controlled injection of microbes in two major landfill sites, which produces huge amount of methane in Kolkata and in Guwahati. And what we have able to show is, top right hand side, if you look at this graph, between 50 to 100% removal of the methane emission post the application of the injection of these intelligent microbes, and all of these are indigenous microbes. So essentially, this system holds for about 6 to 10 months, so we can actually volume wise remove the methane emission, and then it can be then linked to this the recovery of the precious metals or any one of the other RED and other stuff. So essentially, the landfill sites can become safe, and of course, the release of this leachate, that will be much safer. And now all these learnings has helped us to use again bunch of microbes which are of course indigenous. We have integrated into nanocomposites, and here is one example where we have bioremediated completely 1 kilometer stretch of a polluted river in Assam. And now the river is actually functioning. I do not know whether you see or not that, but the bottom right hand side image of April 2024, the fish population is back, and this is connected. This river is connected to Deepor Bill of Assam, which is an Ramsar wetland of international importance. So it's also sustaining the livelihood of the communities without any other additional intervention. The microbes are driving the what we call it as the passive restoration of the rivers. The top bottom right hand side image is another interesting work that we are doing, where we have put the microbes to remove chromium, which is a major contaminant in one of the biggest wetlands, 40 hectares in Kolkata. It has received for 20 years, leather effluent, and is under the Amrut mission of the government of India. We are slowly restoring this wetland, and what has happened by now is since the intervention, the Putias population, which is a very sensitive fish to pollution, it has returned back. So essentially showing that this can work. Very quickly, the bottom right hand side image is another example where we have used these microbes in Leh and Ladakh sector also to tackle water quality on a large scale. I will now shift, because of lack of time, that this knowledge of microbes are not only restricted to urban areas. We have now used these microbes in a combination with indigenous oyster population to slow down coastal erosion in mangroves, particularly from Sundarbans. Professor Krishna Roy talked about it, and it has shown remarkable result where we have more or less managed to hold the islands where there was high rate of erosion using the combination of these microbes and the oyster population. Last but not the least, I want to give an example very quickly where we have used the microbes in shrimp ponds, and we have brought in this concept of mangroves with shrimps. Essentially, the farmers are no more using antibiotics, no more pesticides, nothing like that. The shrimps that are growing are completely healthy, organic, and now they are getting exported to Europe and Japan. Again, using microbiome learnings from the field. And I'm very pleased to share that this has received the Global Technical Award of 2025 from the Food and Agriculture Organization of the United Nations. I come and end the last slide, which is the bioeconomy driven slide, of course, with the connecting with the previous one. And here is one example where the indigenous cyanobacterial population has been used, scaling up has been done up to 1,000 liter to produce dolastatins. Dolastatin is a very important drug scaffold for tackling cancer, So we are able to do that, and we show that this could be a very sustainable method for actually producing bulk volume of dolastatins, which is an immediate need for the pharma industries, and they are desperately looking for it under the Atmanirbhar Bharat scheme. What I want to highlight is that DBT has very generously supported us. Our learnings from the microbes and the nanocomposites, now we are able to show that it can remove 50% of the common microplastic pollutants, particularly PET and polyethylene, under control conditions, and we hope that in future we'll be able to use these learnings to apply in the field under control condition. We are very thankful for DBT to supporting us, and also our lab members who have done all the work. Thank you, sir, for this opportunity.
Thank you very much, sir. Thank you very much to take all of us and all the participants through one of the universe of different activities, and it's fantastic to connect how microbes can be used, or how these microbial cultivation, algal cultivation methods can use and can contribute towards the larger large scale global problems. So really, this can be helpful in addressing environmental issues and climate change issues. So thank you so much for connecting us with this all. So now we are moving to number 10. This is dry times at forest ages, seedling traits and response to drought. Dr. Meghna Krishnadas from NCBS Bangalore. Can you hear us?
Good afternoon. Yes, I can hear you. Can you hear me?
Please go ahead. Yes, we can hear you. Please go ahead.
Okay, let me just share my screen. Yes. I'm sorry, just give me 1 second. Can you see my screen?
Not yet, ma'am. Kindly go to share button in bottom.
Yes. Panel. Can you see it now?
Yes, perfect. Oh, please go ahead.
Okay, thank you so much. Well, first of all, thank you so much for this opportunity to present my ongoing DBT project, which is to understand how forest regeneration varies in response to potential changes in the climate and in human modified fragmented forests of the Western Ghats. Thank you so much to DBT for funding this work, and we are pleased to say that a lot of the work that has gone on is already showing successful results. So to start off, just to set the context, in today's world, human activities have altered ecological processes and ecosystems at the planetary scale. And these massive global environmental changes are having effects on different species and ecological communities. And a major scientific goal is to understand how species are going to respond to these changing times. Now, while individual threats such as habitat loss or climate change have seen a lot of work, what still remains understudied is the interaction between these forces of change that ecological communities are experiencing. So this forms the crux of the project that we are trying to do, which is to understand that if you have climate change, which causes issues like drought in the future, how will that react with another problem such as habitat fragmentation, which creates these edges that disrupt what ecological communities look like. So let me just explain this to you. When you have a large patch of forest, any patch of forest will have some area that is comprises of the edges. But when you break up a large patch into smaller fragments, you increase the proportion of edges that are there in that landscape. Now, edges tend to be warmer and drier. They're more subject to desiccation. So logically, we would think that with climate change, with there being a drought, edges of the forest are going to respond or be affected differently from the interiors. We can also think of this in a way that species that are, that can colonize at edges will now be restricted not only by the local changes, but by the drought effects that might happen, and forest interiors might serve as refugia where some kind of species can persist only there. So we wanted to understand some of these from the aspect of species functional traits, which are heritable characteristics of their leaves, their stem, their roots, their physiology, especially for plants, which determine how they use resources and respond to different stressors like drought or other changes that might happen. And we wanted to do this in an experimental framework by setting up a potential drought situation in the field itself. So this was an ambitious experiment that we were trying to do. We went to a forest. We've been working here for almost a decade now, so we know the landscape very well. And we set up these experiments, if you can see my cursor, where we had these paired sites at the forest edge and the interior, where we transplanted seedlings of 16 different species that we grew over a period of 1 year from seed into seedling in controlled greenhouse conditions. And then one of these treatments, we imposed a drought shelter, a rainout shelter, which reduced the availability of soil moisture. And because we've done pilot studies before, we know that soil moisture decreases considerably with these drought things. And even in this study, we see that the response of soil moisture becomes most prominent in the dry times, which supports one of our hypotheses that forest edges, when things get drier, edges are likely to get more dry than interior forests. Now, first of all, when we measure all of these traits among these species, we find, interestingly, that the water use traits, which is traits related to stomatal anatomy and turgor loss point, which is a key physiological trait, and other anatomical traits related to water use, they form a completely different dimension of species strategies as opposed to the morphological traits. And so far, most studies have only focused on morphological traits. So we found that this understanding water use traits is seemingly really critical for tropical forest seedlings. And in that, mostly we need to look at this one trait, which is called turgor loss point, which is basically the water potential at which species start wilting, the leaves start wilting, a key indicator of their drought tolerance. The more negative this trait is, the more tolerant a species is of drought. So we have characterized this, and we see that even among what are called wet, humid forest species, there is substantial variation, which is a very important insight that we have gained, which means that species will respond to drought differently based on their turgor loss point. And by the way, this is all data still being collected. So what I'm presenting here is quite preliminary. And in a future presentation, I hope to have more robust results. But even in these preliminary data, what we see is that species hydraulic safety margin, which is basically the difference between the minimum water potential that they experience in the soils that they're in comparison to their turgor loss point. This is a great indicator of how well they are doing in drought. The lower it is, or the closer it is to zero, or if it is negative, it means they are under drought stress. And very clearly you see that regardless of whether they are at the edge or in the interior, the drought treatment that we have imposed pushes them closer to their safety margins, which means that they are experiencing drought stress because of a loss of soil moisture. But this differs among different species, as you would expect, because they differ in their physiological tolerance to water scarcity. But most importantly, what we find is that the closer they are getting to their safety margin is actually more intense when you have drought in the forest edges, which is exactly what we hypothesize, that edges will make species more susceptible to drought, which means we need to take these steps to conserve species at the edges because they are being more susceptible to drought. We see this in species responses in terms of their mortality as well. There are some species like Holigarna nigra and Symplochos racemosa, which are showing much higher mortality in drought compared to controlled conditions and more prominently at the forest edges. But of course, there are some other species where you don't see that much of a difference, which might be related to their hydraulic safety margins or their turgor loss point, as I mentioned. So these were preliminary results. The next steps are that we want to have these more robust statistical models to understand species drought responses. And these responses will be in terms of their survival, as I just presented some data here, their physiological responses in terms of how much they're photosynthesized and the growth of their biomass. We also want to understand whether it's individual traits or the whole plant phenotype that explains drought response, and whether this explains the difference between edges and interior. And why is all this important? Because this is the UN Decade of Restoration. There are massive efforts to repair degraded forests using species, but we don't know which species are good at in different microhabitats of these degraded forests. Understanding how species might respond to drought and microhabitat conditions is going to help us plan better for restoration by choosing appropriate species. And the next step, of course, is using all of this data to make some predictive models of what the future seedling community is going to look like, for which we have already initiated some collaborations with modelers and something that my lab also does. And with that, I would like to acknowledge my student Gayatri, who has been leading this project in the field and doing a fantastic job. But along with this field team, which I'm very grateful to, this is a lot of hard work, collecting seeds, growing seedlings. It takes a year before we even start the experiment, and it's a lot of hard manual labor. And these people have done an excellent job, so I thank them for it. Of course, Department of Biotechnology for funding this project, and TIFR and CPS for the support that they've given in terms of logistics and others. With that, I'm happy to take questions. Thank you so much.
Yes, thank you so much, ma'am, for sharing the fantastic insights and how you are working to define the the traits on the edges or forest edges, and how it can be important to establish further the predictive models based on this. Really, once it is established for one, definitely it will work for other traits also, and it will lead to somewhere to some important solutions from climate angle. And really, water stress is one of the major issues that is happening because of the climate change, and this has to be addressed in our forest. And forests are obviously very much important to mitigate climate change further, or towards consolidating our mitigating efforts. So forests are almost on edge of the climate, we can say. So those edges
Yes.
has to be defined and addressed. Thank you so much, ma'am.
Yes. Thank you so much.
So
Thank you.
now,
Yeah.
so now we are at serial number 8. This is pollinators and ecosystem services in the Anthropocene. And the speaker here is Dr. Hema Somanathan from Indian Institute of Science, Education and Research, Thiruvananthapuram. Thank you so much, ma'am. It's over to you, ma'am. Please finish in 5 minutes.
Can you see my screen? Hello?
We can't see yet. You had not
Not
shared
yet.
yet, ma'am.
Okay, let me try once again.
Just go to share button in bottom panel.
That's what I did. Share. Entire screen is what I selected. Can you see it?
Share once again. Share once again. I think not yet shared.
Selecting entire screen, she has to press the share button.
Oh, after selecting entire screen, then again you have to press the share button.
Yeah, the share button is not active over here. Only the cancel button is active. Okay,
I think some pop active in your. Yes, yes, yes. It's working now, I think.
Yeah, is it okay? Can you see it?
We cannot see your slide.
Oh, okay.
I think it's going right way now. Huh?
Entire screen, but the button at the bottom doesn't become active. The share button is not active.
You need to select the screen.
Sorry?
You need to select the screen. Click on the screen which is coming before going to the bottom of the button at the bottom.
Okay.
Entire screen, then select the screen, then the bottom button will get activated.
Okay.
Entire screen.
Yeah.
Entire screen, then a screen. Yes, we can now see. You can go to yes.
Yeah. Okay. Yeah. So at the outside, thank you very much. Thanks to DBT for this opportunity to present the work that we've been doing, which is funded by DBT under the Forest Biotech program.
So in this program, in this project, what we have been doing is to look at pollinators in different landscapes across 6 Indian states and try to quantify pollinator diversity as well as ecosystem services in terms of pollination to wild plants as well as crop plants. So since there is no time to get into the details of our results, I'll just briefly take you through what we have been doing so far in the last 2.5 years. So basically, I mean, as
5 minutes
all know
only,
and
yes.
appreciate, pollination is an important ecosystem service, and a lot of wild plants are dependent on animal pollinators, as well as a lot of crop plants are also dependent on animal pollinators. And so therefore, pollinators play a crucial role in biodiversity conservation, food security, poverty elimination, and
Dr.
so on.
Hema, since we are short of time, we'd request you to just present what are the highlights in just 4 to 5 minutes.
Sure.
That would we'd appreciate
Yeah,
that. Thank
that's
you.
all. I mean, I'm not going to take more than that. Yeah. So these are some of the food crops that are pollinated, out of which we have focused on a few of these plants. In so that
Is breaking. There is some connectivity issue, I guess.
We cannot hear you, ma'am, actually. Acha.
is a western honeybee, but that's an exotic species.
Please go ahead.
Ma'am, I think we can't see your screen and you are not audible. You need to unmute.
Can you hear me? Hello?
Ma'am, can you hear us?
I can hear you clearly. Yeah.
Yeah, your share screen has stopped. You need to reshare the screen.
Can you see now? Hello?
Please go ahead, ma'am. Yes, we can see.
Okay. So I will
Again, some
talking
connectivity
about,
issue is there.
using the waggle dance of bees as a tool in order to try and see here is a
Ma'am, sorry,
yeah.
I
Hello?
think there is some connectivity issue from your side.
Hear me?
There is issue with internet. You had been logged out earlier, and now
Hello?
again the slide sharing is not happening. So in between, you can work out with your system, and we can take another presentation if you don't mind, please. Yes. So now we are at serial number 9. This is invasive alien species and their threats to native biodiversity in Ramsar wetlands. Here the speaker is Dr. Ravikant Godasala Mani from BioE3 Bangalore. Sir, we request you to kindly share your slides and finish it in 5 minutes. You also left. Possible to you to share your slides now, ma'am? Dr. Hema Somanathan, ma'am, would it be possible to you? Can you hear us?
Yeah, I can try. I can try again. I can try again. Yeah.
Can you put two of your slides about the achievements? Those you can share. Only two slides where achievements are there.
Yeah.
So just share those and then we can bind up, I think.
Yeah, sure. Can you see my screen though?
Not yet. Yes.
Okay. You can see my screen. Okay, fine. I can hear
you need
you
to
all.
go to.
Yeah,
We
my
can see your
presentation.
screen. You need to go. Yeah, you need to go to the
Yeah.
presentation mode.
Yeah. I'm going to my presentation. Yeah. I don't know why my presentation has closed down, so let me just find it. One second. Yeah. So this is here is, I will probably wind up in 3 slides. So can you hear me now clearly?
Yes, ma'am. Perfect. Please go ahead.
And you can see my screen. Yeah. All right. So basically, just to give you a sample of what we have done, we actually had these feeders which were placed in the landscape in different locations. For example, what you can see here is rubber plantations.
Ma'am, I think there's some continued connectivity issue. So, yes, I'm sorry. Actually, we can take then another speaker, and you can send the recorded presentation so that we can, share with the relevant participants. So now we are at serial number 9. This is invasive alien species and their threats to native biodiversity in Ramsar wetlands from Dr. Ravikant Godasalamani from IIT Bangalore. It's over to you, Dr. Ravikant. We request you to kindly finish it in 5 minutes only.
Yeah.
We are
Can you
beyond
see
the
my
time.
slides? Can you see my slides?
Yes,
Hello.
yes, perfect. Please
Yeah.
go ahead. Please go ahead.
Good evening, everyone, and thank you for this opportunity to present the work that we have been doing. This is about mapping the invasive alien species and their threats to native biodiversity. As we know, a lot of flora and fauna are threatened by a number of invasive species, and especially so in the biodiversity hotspots, especially in the Ramsar wetlands, where both plants and animals have invaded these Ramsar sites and have been destroying or having are major threat to the biodiversity in and around those wetlands. Some of the Ramsar sites are also, especially what we have been working on is the number of fishes that have actually got introduced into the Ramsar wetlands, either deliberately or by accident, and they have caused a large threat to the native biodiversity. In fact, many of these invasive species have both economic impacts because of their presence and because that they are feeding on the local biodiversity, but also there is a lot of cost with respect to their managing these invasive species. Apart from that, we also see that because of the presence of these invasive alien species, there is also a livelihood that is threatened because these invasive alien species or fishes are also causing problems to the native fishermen who are fishing in these wetlands. So our objectives in this particular project has been to determine the occurrence of freshwater invasive alien species using a non-invasive method, using a eDNA method, and assess the dietary composition of some of the invasive alien species, and then develop a management plan integrating citizen science approach. So one of the things that we have been doing is also developing methods for eDNA detection. In this, one method that we have all tried and have developed now is to develop a low-cost eDNA kit for detecting, for not only sampling water, where we can sample 5 to 10 liters of water in a filter system. And this is a low-cost equipment that we have been trying to develop, which can also be carried to different field sites and then use this to capture the DNA from the water. And then we are also in the second objective, we are trying to capture the invasive species, extract their gut content, and then do a metabar coding to identify what are the impacts they're causing to the native biodiversity. And third is the community engagement, where we are training the local fishermen and other youths in the neighborhood to identify these invasive species and then establishing a map and trying to help them manage these alien species. So this is a low cost machine that and a handy machine that we have developed with the company, where it can be easily carried to the field, and which operates in a small pump, the battery, which is less than 10,000 mAh, which can be taken in the flights as well. And what we have done is now mapped some of the alien species in the Ramsar sites, and we are trying to now use eDNA methods to actually quantify and see whether many of these species are present or not. In one of the first, this is just the first year of the project, where we have now done in Dipur Bale in Assam, and in Tripura, where we have looked at the Ramsar sites there. And what we have found is in both these wetlands, we have about 5 to 6 invasive species that are present, especially the African catfish, which is causing a major threat, has been found in both these wetlands. And the other thing that we have done in a preliminary study, what we have also seen is that, for example, one of the species, Gambusia, which is an invasive alien species, we found that in the gut content of this particular fish, a large number of native fish DNA was found, apart from the mosquito larva that was actually intended to be there in this fish. Actually, the species was introduced to control mosquitoes, but that mosquito diet was only 2% of their diet, and the rest 98% seemed to be other flora and fauna that is existing in that particular wetland. So now what we have done is also develop some modeling approaches to map many of the alien invasive species across the wetlands, not just restricted to Ramsar sites. And we are also creating awareness among citizen science about these invasive fishes, which can cause major havoc in the Ramsar wetlands. Thank you, and this is just the first year of the project, and we are hope to continue to do this across other Ramsar sites. We'd like to thank the Department of Biotechnology for giving this opportunity and also for funding this particular program, and Dr. Robin Raja, who is leading this project as part of this study. Thank you very much.
Thank you so much, sir, for sharing these insights on alien fishes and native biodiversity threats. Obviously, ITRI is the very important stakeholder for all the biodiversity related problems, and biodiversity conservation is very much linked in both ways, either for threats due to climate change or for action towards the climate protection. So that biodiversity conservation or management, we can say, that is very much important, and ATRI is contributing very much towards this cause. So thank you so much for sharing the insights. So ma'am, we are done with the technical program. We can take some QA if there are some questions from online participants. 1-2 questions only. So there is one question from online, from YouTube, to Dr. Azaj Ahmed. The question is, can biobutyric acid and biobutanol can be considered to use in SAF production? Dr. Azaj Ahmed, sir, it's for you. I think he's on mute or.
Pass on these questions to respective speakers.
He has gone out for a minute. He will be here in a minute, ma'am. Professor Ijaz Ahmed will be here in a minute, ma'am.
We can wait for 1 minute. Is there any other question from any of the participants?
Yeah, sorry. So I went for water.
No issue, no issue. So the question is, can biobutanol and biobutyric be used for SAF production in coming days? So this is from participants from YouTube. Biobutyric acid and biobutanol can be considered to use in SAF production.
Yes, so exactly this is what we are doing. So when butanol is produced, butyric acid comes as a byproduct. So we take them as of now, both are getting separated. But if we talk on a broader perspective, we can use a catalyst in the same ferment mixture and we can make the butyl butyrate. And butyl butyrate, we have already showed it can be used up to 50% for blending.
Great. So one more question is there. This is very general question. So he says there are various efforts being made under research and development for biodiversity conservation and forest protection, as well as other conservation initiatives. So why the achievements made in the laboratories are not coming to the in kind of awareness in general public? So that is kind of concern. So can somebody comment on this?
So I would say this is a very good question. If I answer from my perspective, the real challenge is when you develop a technology in the lab. After that, it requires a lot of people from mechanical, electrical, electronics as a EPC consultant to scale up. And right now, that link is missing. So we develop the technology, but for a scale up, we need a bigger team to come together.
Any other question? If not, there is one suggestion from somebody on YouTube. So they say, let's promise to plant more, waste less, and love the earth. So with this, the program is over to you, ma'am, for your closing remarks.
I would congratulate all of you for the excellent research and achievements what you are continuing, and would wish you all the best for future. And what is important that we need to translate this research. So in the field, and most of you are doing, so there are some few examples which I have seen, like mangroves and all those are happening in the field, and which is really creating the impact, and that has been demonstrated. So request all of you, all the project investigators, once again, thank you all of you for joining and presenting your work in very short time and short notice, and with enthusiasm. I think he has written the poem. That is also a compliment to Dr. Balendra for the, he has been writing poems on this, and we all are, together we can create real impact towards the sustainable development and how to protect our environment. With this, I would thank all of you once again, and over to you, Balendra.
Thank you so much, ma'am, for giving this opportunity and inviting all and providing this platform to share the thoughts and developments happening in the different programs under your esteemed guidance in forest biotechnology or advanced biofuels under the Ages of Biomanufacturing program. I also thank special thanks goes to today's chief guest, or we can say guest of honor, Dr. Anil Prakash Joshi, Padma Awardee, two-time Padma Awardee, Padma Shri, Padma Bhushan, and one of the great environmentalists, for telling us about the impact of the smaller initiatives and how they can lead to big changes from climate point of view, and how our small efforts at individual levels like carbon footprint mapping and this can help in building the resilient ecosystem. And he has delivered it in the manner that one common man can understand. That's very important. I thank you on behalf of this program division and Department of Biotechnology. The special thanks also goes to Secretary Department of Biotechnology for allowing us and guiding us with its leading remarks how the further activities can under biomanufacturing can provide and enlighten us to work further for development and expansion of different programs on the lines of national priorities under climate change. I thank each of the investigator very much for coming on this platform on very short notice and sharing the insights. I cannot explain each of that, but we have received very good comments. I mean, very good comments on the YouTube and as well as from online participants. Each of your lecture and each of your slide, whatever you had delivered here, and many of the students had commented there. So thank you very much for sharing these insights. These will help, somewhere transforming the programs being built by Department of Biotechnology. Thank you so much. We will work together. Thank you for all the online participants who have joined either through virtual link. Thank you for MOAFCC. They have, delivered the converging policy remarks, how the government of India is making the wholesome efforts towards climate change mitigation. So thank you all of you again. Thank you very much for all participants, attendees, students, teachers, faculties, everyone who has, I cannot name everyone, but everyone, thanks goes to everyone. Thank you very much.